Okay.
So we are 1,461 meters on the unnamed seamount, 76 degrees north, and we're 3 scientists here in the shack. Annalena from Seckenberg.
And Paco.
Myself, Paco from
Uppsala.
Museum of Evolution at Uppsala University.
And I'm Jenny, also from the Senckenberg Society of Nature Research in Hamburg.
Yeah, can we get a decimal move, please? Bearing 125 at 0.1, please.
So we just landed on this seamount, which is on the eastern side of the Arctic Mid-Atlantic Ridge. So during our previous dives, if you followed them, so yesterday we were, for instance, at the Schultzbank seamount. We were on the western side of the Mid-Atlantic Ridge, and now we're exploring a seamount on the eastern side. And this seamount, although it's been mapped, it's never been explored. That's my understanding.
As far as we know.
Yeah. So we're the first humans to actually visit it and see what it looks like. And of course, there are loads of sponges. But not only that, I think we've already spotted a few fish.
Like this one.
Like this one.
And Lysodendorix.
Lysodendorix, the sponge, the pompon sponge. We have 2 lasers at the center of your screen. They are 20 centimeters apart. So they can give you an idea of the scale. And just before we started going live, we heard the news that we had other animals around the boat, right?
Yeah, we believe we saw northern bottlenose whales that were
feeding, maybe.
There were several, and we've been observing them for about an hour now.
I think there's a team outside with a drone trying to get some footage of these whales, so that's pretty exciting.
And if I'm not mistaken, these whales are amongst the deepest diving whales that we know so far.
That's true, and they feed on squid.
And they feed on
Yeah, so
squid.
Maybe we'll encounter some.
Wouldn't it be cool to see a whale, though?
That would be something.
But let's see, they probably heard us from far away.
Yeah. Let's get some anemones in the log. Anemone and Lysodendorix, Asbestopluma.
What Jenny is naming here are sponges.
Sorry, yeah.
Paco, are all these potato-looking unit sponges?
Yes, there's all of the potato-looking spheres are geodias, most probably. But I cannot tell you the species unless we zoom a bit. But there are only 2 species in this area, Geodia parva or Geodia henscheli. These are the 2 Arctic Geodia species. These are massive sponges. They can get to sizes up to 30, 40 centimeters in diameter. You can see here with the lasers that these ones are about 20, 25, which means they're already quite old, actually. They might be already 100 years old.
We're moving up.
Yeah, let's do that.
So why are they covered in sediment?
They seem to be on the cliff, and there might be a lot of sediment deposited on the cliff. I'm not sure.
Oh.
Yeah,
It will regrow.
Yes.
It will settle.
Sponges are really remarkable. That's more for Paco to tell you about sponges, but I'm so impressed that they regrow from small pieces.
Yes, they can regenerate. They heal very rapidly.
contains still.
in 1 or 2 days. I did some experiments actually, especially in the tropics, where I went on the reef, took a piece of sponge,
Bridge, can we come to an all stop there, please?
and then revisited the same sponge the next day, and it had already healed.
Thank you.
So
Wow.
yeah, and then they can regrow as well, quite fast. So that's the advantage of having absolutely no organs and very little tissue. It's essentially cells moving along with bacteria. They are living in symbiosis with a lot of bacteria.
Mm.
Something wrong, I cannot forward.
No.
The sea star will settle again too.
It's not around 10. Don't go so close, because the back generally into the ship moving.
No.
No,
Okay.
because the track is like this.
Yeah, it's alright, yeah. Just make sure you go to the one to the southeast.
Oh, we just need to get a bit into the groove here because the slope's very steep.
Yeah,
Sometimes when we
No, if I
plan,
go up, I go under the ship.
when
It's
we
alright,
plan
just maybe
these
just step the ship back
transects,
if you have to.
it's very hard to estimate how it
So
will
we
be
don't
when
follow
we
the
reach the
track,
bottom.
or
No, you
it's
have
two
to.
different things.
because
That's-
of course when we say that
No, if I go up, I go under
the
the
seamount
ship.
is
That's fine,
mapped,
but move the ship
I
back
mean
if
it's
it's
not a
cheap.
very precise map. It's a very rough map, so we don't really know the details.
No, exactly. Is that a sea urchin?
So eventually you will be creeping forward as you come up,
Yeah.
as long as we're
I don't know.
roughly coming to the southeast.
No,
No,
it's
that's
an
an
anemone.
anemone.
Yeah. That's an anemone.
1,400. Yeah.
And the sea
Starting to pop
star.
up there gradually, yeah.
And there's a fish hanging out on an asplastopuma. You see?
Oh yeah.
It's really looks like it's sleeping on it.
Mm.
I know I'm doing fine.
Okay. Shall we stop?
Do you want to stop?
No, I'm just asking those two guys first. Shall we stop and able to zoom?
Is that a
Um,
possibility first, or is this
Yeah,
tricky?
to stop?
Do you want
Just
we
here,
can check
just here
yeah.
we can.
Yeah.
Okay.
If you can hang,
Not too
we can
close,
hang here,
so
not
that
to land, but just to stop.
yeah.
just to get an idea of
With
the
the shrimp?
fauna. Yeah,
Exactly.
yeah.
And all
Okay.
of
Thank you.
we're done, so we can zoom in just. We see some worm tubes again,
Yes,
polycate worms.
absolutely.
And the white spot is a strong now moving out of the
I
picture.
think these are the ones our colleague Sergi
Mm.
Taboada would like to collect.
You didn't understand
No.
what I was saying.
I'm not sure. Yeah.
Is there a possibility to collect on the cliff, or is that too complicated?
What do you want to collect?
Yeah.
Are you going to? Do you want to slurp?
Yeah, with a slurp.
No.
No.
Sorry.
Okay. No.
We're right up against the cliff.
Okay. Yeah.
Okay.
No, let's
So
try to
let's-
find a better suitable place.
Oh,
Okay,
sorry.
let's
You
zoom out
can
in the front.
see-
Yeah.
That's yours.
Sorry.
Let's zoom out in the front.
Yeah.
We
Just
can
wanted to have a quick look.
we can log that we have polychaete mats
Yeah.
on the cliff. We can we can continue the climb.
This.
Yeah. I know we can turn around.
Well, we are not climbing. We are just going laterally
Right.
around the wall.
Right.
The wall.
Right.
Yeah.
Okay.
That's all good.
That's
Yeah.
perfect. Thank you.
Andrea is our ROV pilot today, and Rob is our co-pilot. So we of course we listen to them very carefully because they know what the robot can do or cannot. We can see a lot of what I call pompon sponges. These little bushes, white bushes, This is a species of sponge called Lysodendrorix complicata. And then we have the large vase sponges, which are glass sponges. These can be quite big, a meter large easily.
They also occur in the Antarctic.
So in
Okay.
the Antarctic, you find exactly the same family, yes, and they're even bigger in the Antarctic.
Do they also occur in other oceans?
They do, but I don't know if they reach those sizes, and I don't know if they dominate like they dominate here in the Arctic. So my guess would be that they will become more and more present as we ascend this unnamed mountain.
So
We're currently at 1,455 meter depth.
Mm-hmm.
there's something. Something was looking at us from this upper part there. Probably a fish, but you know, since you said that the northern bottlenose whales feed on squid, I'm like, maybe we'll spot some.
All the little white tree-looking structures are also sponges, but they're carnivorous sponges, meaning that unlike most of the other sponges who are filter feeders, they filter the water to eat, and they eat very tiny things in the water like bacteria or organic matter, particulate organic matter. Carnivorous sponges are actually going to eat little animals, like little crustaceans, like little shrimps, that they're going to catch, that are going to be trapped on these little trees, and they will be digested by those sponges.
There's an interesting black fish, an eel pouch
Yes.
kind of fish.
Yes. So we write everything, or the highlights of what we see, because the recordings of our exploration will be used later on.
Look at that camera. It's a whole.
Oh yeah. So it's like a
It's amazing.
very steep cliff.
It didn't
Yeah.
look that steep in the planning. It's very interesting how it's.
Maybe it's a very also localized cliff,
Yeah.
and you don't see it on the map.
So we have several camera views here in the shack, not only that one that you're seeing. And on one of the cameras, we see that we're currently flying above a gorge. Like it goes really deep down just below us, which is pretty cool and exciting.
Okay, cool. Bridge RV, can we continue the move, please? 125 at point 1.
We constantly need to tell the ship
Thank
what
you.
we're doing, because we move in tandem, so we have to move together because we're connected through a cable, and we are very, very deep below the ship. So whenever we go forward, the ship follows us, and when we stop, the ship has to stop.
Can you log amphipod?
I can. I will do it if you explain to the audience what an amphipod is.
That's Annalina's speciality.
They're the best.
Okay.
No, that's all.
That was short, but
Okay. Well, amphipods are little crustaceans, also known as beach hoppers. They're dominant. They're super important for all ecosystems here, for all depths. They can be found in freshwater, in caves, and in the ocean. From shallow beaches down here to the Hadal trenches. So they are really essential.
is it a large group? In taxonomy, how many species are we talking about?
Around 10,000 I know now.
Are they mostly in the deep sea or in shallow waters?
No, they occur in all depths, but they seem to be more dominant in the deep sea because not many other animals
Mm.
can survive, for example, at high pressures of trenches and canyons. So that's the only type what's left. And the more shallow we go, the more other invertebrate groups are to be found.
The anemones are so beautiful.
Sorry?
And there gets more.
This is all that I was trying to explain.
I'm going in the trunk.
Oh, look at these masses of geodia. So these are very big, because if you look at the laser, it's 3 times, so maybe 60 centimeters.
Which is enormous.
Group RV, can we stop the move, please?
That's what they want. Otherwise, we're just going at the same depth. We're not coming up there.
These are the first images of this seamount.
Yes.
Wow.
This is a slightly challenging place to start a dive,
Definitely.
but where,
we didn't know. Yeah.
because it's very abrupt, but it is very rich, and probably these two facts go together, because usually when you have cliffs, you have a lot of currents, and if you have a lot of currents, you have a lot of food coming in, and many of these organisms rely on the food that's in the water column, so they will actually like to settle on these cliffs.
And they do, a lot of them have larvae for distribution. So they need something hard to sit on. and
Absolutely.
grow. So
Yes, you're
that's
right.
That's why they also need hard, so-called
Hard substrate. Mm.
And a rock, of course, is a hard substrate.
Bridge RV, can we continue to move, but instead of 0.1, can we go 20 metres at a time, please? Lovely job, thank you.
It's very beautiful in a way.
And
because
and I don't think we've ever seen such such a
no,
type of
no, this
community.
is-
It's
this
yet
is
another
new.
one.
Yeah.
So, do you think that all of these dark patches are the worm tubes?
So yes, yesterday we discovered some walls covered with worm tubes, something we had never seen before, a community and a habitat we had never seen before. And here they look, I would say yes, it looks like patches of that
Coming in.
type of habitat.
Yeah.
But we may need to look a little bit closer.
Yeah. I just don't want to zoom in too far
No.
because it gets
Hard on the eye.
hard on the eye. Thank you. There's again a fish asleep on one of your asbestoplumas.
Mhm.
Excuse me. Are they using this as a mattress, or
I don't know. Maybe
they're sleeping on it?
Maybe they are. Why not? Sponges are supposed to be comfortable.
I mean,
Well, we touched one in the lab today,
and
which
this
was
one is-
surprisingly soft.
Yes, that's true. And
So,
these are,
well-
it was one of those big, large white ones.
Mhm.
And yes, I agree. I was not expecting that it would be so spongy, actually. I was expecting something more brittle and fragile. But it was very thick and soft.
a spongy sponge.
Mm-hmm.
There's another fish. It's quite rich here. See? It's lying
Yes. Let me actually.
there. That
put
might
this.
I think it's another feather star there sitting on the sponge.
Oh, it's the first feather star of the day. There are more to come.
Yes. So definitely more fish than yesterday.
Yeah. Yeah.
So we are on the eastern side of the Mid-Atlantic Ridge,
look
and
at
so we're very interested to compare with what we've seen on the western side. Because the Mid-Atlantic Ridge, as you know, is a border between the Eurasian tectonic plate and the North American tectonic plate. And so far we've been diving on the North American side. Today we're on the Eurasian side, and people that study biogeography of animals, meaning their distribution, always wonder if you have the same animals on either side of the Mid-Atlantic Ridge, because it is quite a border. It's quite deep, so it is quite a barrier, maybe for many larvae or small animals. So this is a little bit the incentive to come here to this eastern side seamount. Never explored before us today.
That, I believe, is an hormatiid anemone again.
And Nina, do you have samples actually from that side of the Mid-Atlantic Ridge?
No, I don't.
Me neither,
That'd be
actually.
great.
And another feather star.
And the fish as well.
And wow, the crinoids are incredible.
Oh, there's a very nice one. So crinoids are part of the sea star family, right?
Yeah, that's
Like sea urchins.
not the sea star family.
No, the family.
No, the the phyla, the
phylum.
kind of
Yeah.
the group. Sorry.
No, I'm sorry. There's a tilasta in the wall. That's a sea star.
Yeah.
They always look a bit chubby.
Now what's coming up here?
I really like them.
Huh?
Make sure you keep it
Yeah.
tight.
I like that structure.
Can we hold for a sec?
Yeah.
Thank you. Beautiful. And there you can see on the rock, you see a couple of white tubes, and those are the calcified tubes of bristleworms. And
all of them that you can see are filter feeders. So the feathers are, this is arms out. Small, see there, on each arm? They're all trying to catch things in the water column. So all their growth of all these animals relies on other little animals for nutrition in the water column.
And I can now tell you that the Geodia is Geodia parva, because I can see the openings on the top surface, so I can now identify the species. But I'm able to do that because we have been collecting it before several times, so I know now exactly how it looks like. Otherwise, it would have been impossible to identify it. So that's why we're collecting more and more as well, to get a better understanding of the biodiversity and the species we find here. Look at all the tiny sponges also on the ridge of the rock.
I would zoom back out to continue.
Yes.
Is that all
Sure.
right?
Yes, absolutely. You see a lot of carnivorous sponges, those little white bushes or tree-like structures. These are sponges,
Yeah.
but carnivorous sponges.
I can fly on.
Yeah, I'm just going to turn my head to starboard. I want to see where that line
Sure.
goes in.
Sure. It's the folder here actually.
This? Oh no, the folder, the master folder. This one.
The master folder. I am interested in whether the transects have made it here.
So while we
Sorry, I'll show this on 20.
turn around this cliff,
Oh,
Copy that, thanks.
I can say that we are 6 scientists on board of this deep Arctic expedition.
So keep the ship there.
We left Ireland the 8th of May, so we've been in this boat for nearly 20 days, exploring the mid-Atlantic ridge in the Arctic. Seamounts, rifts, hydrothermal vents, volcanoes. What did you say?
Coral gardens.
Coral gardens, absolutely. That was our first dive. So if you want to see any of that, please watch our earlier dives.
Crinoid City,
Crinoid City.
Anemone Alley. I mean, come on.
Reach ROV, can we get a vessel move, please? Bearing 230 for 330 meters at point 1.
Soft
I
coral.
think it's a
Yeah,
soft coral.
exactly. Our first soft coral.
Andre, look how undramatic this looks.
Thank you.
Thank you for the resolution.
Not yet.
35.
Yeah, see the way it goes.
But it looks so undramatic. It's a great, yes. Incredible.
It's better like this.
Yes.
Jenny's comparing
So,
the Transect that was prepared with this. It just looked steep.
it's like
20 meter range.
we have maps
Yeah.
because scientists have been here before and mapped the seafloor
From the surface.
from the surface. So we believe no one has been down here with an ROV. We are the very first ones to see these animals and to see this ecosystem. But this area has been mapped from the surface and the resolution is pretty good. However, I would say that the transect sort of the topography of the transect that we planned is nothing compared to what we see here, which is very complex and steep and rugged. So you never really know until you're down there and see it with your own eyes.
And that's why we need to explore,
Yeah, you really do.
because now we know.
Yeah. Would you mind putting it back?
No.
Thank you.
No worries.
Oh, another soft coral. That's nice. So there's for sure plenty of current and food here along this incline, like along this cliff,
Mm-hmm.
because these soft corals need it. Your sponges need it. The feather stars wouldn't be here without food. So it's a very rich environment again.
Mm.
Look at all the geodias.
Mm.
It's really pretty. Another sea star. It's really beautiful.
Yeah, it seems the Rosellidae can actually open quite widely
Yeah.
their openings. Look at that.
Yeah.
The
Akituba.
yeah.
Hello.
What was that?
That was a shrimp.
That was a shrimp.
I see several shrimp.
Yeah, we do.
Pink-
There are
pink
many.
ones here.
There are many. I think it's Butocaris.
But do you think they're attracted to the light?
Yeah,
Yeah.
I think so. But not all of them. But sometimes they get also a bit dragged in, depending on
The thrusters.
the thrusters and
yeah.
the currents. And, Oh, that happens.
So since the beginning of our dive, we've already gone up about 50 meters, and we are at 1,415 meters depth. The temperature of the water is minus 0.3.
Ah,
Not the coldest we've had.
no.
I remember on the western side we've had minus 0.6,
Yeah, yeah, yeah,
but still below zero.
yeah. I'm just thinking in terms of water masses here. That
Absolutely. Yeah.
on the eastern side of the ridge, which this section of the ridge is actually called Carnivorous Ridge.
Mhm.
So which is we have Moon's Ridge, and then it sort of goes on as Carnivorous Ridge.
Yeah, after the bend,
Exactly.
it changes direction.
Yeah, and on the eastern side of this ridge, we still have some influence of Atlantic water masses, whereas on the western side we have lots of Arctic, deep, really dense and cold water flowing down.
It doesn't freeze because it's so salty.
We have cold, dense, salted water here. And we're actually not too far. You were telling me, Jenny, that we're not too far from Svalbard.
No, that's right. I checked on the map, and we're about 215 kilometers off the southern tip of Svalbard.
What?
Yeah, that's another decapod. Very funny. It's a bit white, actually they're red, but when they are in front of the lights, they appear very, very white.
So we're just waiting for the ship to come back around, and then we can come back
Sure thing.
closer.
Yeah.
Sure.
So the ship is going that way.
Yeah.
We want to go on this one.
Okay. We'll have some decapod TV, no worries.
Mhm.
Very elegant, huh?
And this will be a night dive. I mean, for us at least.
Oh yeah. Yeah, yeah.
Except if you're watching from another part of the world. But for us, it is the evening.
But the sun doesn't set at the moment.
But the sun, yeah,
It's always light here, which is lovely.
we are beyond the polar circle, the Arctic Circle, so there is no night actually outside. The sun is there all the time.
Yeah, I mean, we were sipping samples yesterday on
Yeah,
deck, and it
yeah.
was 1 o'clock in the night, and the sun was right in our faces,
Yeah.
and it was really lovely. And when we went back inside the lab, I looked at the clock and was like, what? It's 1:15. That was,
Very lucky with the conditions.
yeah.
It's a very smooth sea.
Do you think these are sponges, like the teeny tiny white
Yes.
that are lined up?
Yes, but there are several that I spotted. I'm not sure which species. We would need to do some close-ups or some sampling.
So we will just continue the climb.
This looks like a little bit less steep,
Look at that
but still quite
shrimp swimming towards us. It's
yes.
funny. Looks very elegant with the legs. It's really nice. There are many. I have to say, I think there are many more than on our previous dives.
I agree.
We have always seen them, but
Yeah.
they're quite abundant here.
Let me write that observation. So quite a lot of shrimps or decapods. Ooh.
No, that was also a shrimp,
That was?
just with a little bit of a different style. Up. That was funny. Just
Just asking if he needs to speed up.
Bridge RV, how much is left on that move, please?
With 8 meters.
Okay, thank you.
there's another Tilaster,
Finished.
another sea star in the wall. Do you see it?
Yes, but it took me a while.
Yeah, there's lots of sponges to distract you. So.
Mm-hmm.
I think it's very interesting because when we're that far away from the wall, it doesn't even look that diverse. And then
It's true.
The closer we fly, the closer we get, the more detail there is and the more species we observe.
And it's even more the case when we zoom in. Yesterday we did a lot of that, stopping and zooming,
Yeah.
and then you see a lot of very small creatures, and you can see how diverse and rich these habitats and communities are.
Stop.
Copy that?
125.
Yeah.
Continuous.
Point 2, continuous. Bridge RV, can we now get a vessel move on bearing 125 at point 2 continuous, please?
125.2 continuous.
Thank you.
Yeah.
I find it hard to believe many times when we sit here in the shack and we have our pilots and we know this is happening live, both
Okay, so around 10
this
meters.
is live, but
Mhm.
also that's live, you know? It's
10
like
minutes.
we're down there right now.
Yeah, the ROV's below
It's
us.
just
So
below
10
us and
starts
Mhm.
coming
we're
in.
seeing this.
Yeah.
This is all real. It's really,
We might
it's not
have to,
TV.
you coming in.
And then we
Oh,
collect and we actually
Yeah,
get
he's
the
quite
animals that we've been
That is-
watching.
that is the best, like to actually see.
Yeah.
And they're always much smaller than what I would expect.
Mhm.
But that has to do with, I mean, we have the lasers, so we know the size of them. But somehow it just looks a bit bigger, but that's these effects that you also have in an aquarium, right, in
Mhm.
general.
Yeah, yesterday after the dive on the Schultzbank seamount, we spent a lot of time processing the samples, and that is something you do not see during these lives. Is that we spend a lot of time in the lab, on the boat, preserving, studying, registering.
Well, we take sediment samples and all these little critters, around 1-2 millimeter in size, can only be seen under the stereoscope.
So you've been sorting a lot of sand and mud
Yes.
to find tiny crustaceans. Any nice surprises from yesterday? Did you find animals you didn't expect, or
Yeah, there are quite some, well, I'm not sure
yeah?
what they are, but we also collect a lot for all our colleagues, so we had a lot of little mollusks, like little snails,
Mm-hmm.
I'll send on, and some to a colleague in England, and we have some Taneas, which are also small crustaceans, to colleagues in Poland. We sent them pictures, they're really looking forward to these samples.
Mm-hmm.
And also many of these colleagues, they conduct this biodiversity and biogeography analysis, so it's always nice to have something from this very deep,
Yeah.
those places in
Of
the
course.
Arctic.
Yeah.
Rope shack dry lab.
Yes, go ahead.
You pull out the fisheye, please.
Copy that.
So we're now pulling out the fisheye camera so that the people in the dry lab can have a different vision than the one you're seeing, but more of a 180 degrees vision. to get another type of footage. So you might see it on the screen at some point. This camera sticking out from the ROV. That's the fisheye.
There's a higher density of decapods for sure. Look at them go. Like we didn't see that many on the previous dives.
No.
There are more here.
But where all our colorful animals?
Yeah, they
So they were at 700 meters. Maybe they will appear, but maybe a bit shallower.
But we've seen some really pretty large homatids and anemones already, so that was nice.
Yeah.
I wanted to add, because you asked Anna if we've seen anything interesting or special, and we have found small crustaceans, amphipods with some parasites. I think that was quite interesting.
Yes, sure.
And we had a...
I think actually this is a really difficult question to answer because there is no non-interesting sample. So all the samples have a value, even if it's not first sight
But
obvious that it's something new. It's most likely, but I'm quite careful to say.
But there will be some that bring you more excitement, probably,
that is
because they're
very true.
more
There's
rare.
not a
Or,
last time.
yeah.
That's true. I was very pleased to find a species that Unnamed and I described 2 years ago.
Mm-hmm.
So I described it from south of Iceland, and it's interesting for us to know it also occurs here.
Absolutely.
And it's always nice to find your own species.
Yeah.
Can you log soft coral, please?
I can.
And Lysodendorex. And isn't that Asconema foliatum? Maybe?
Could be.
Maybe.
Could be.
Definitely more shrimps, yeah. They are really abundant. Yeah, I agree with you that everything is interesting in some way, but there are some things that fascinate more than others. Not a soft coral.
Well, this one sponge was my highlight yesterday, I have to say. Paco, tell us about this.
Oh, yeah.
The roots.
Oh yes,
I've never seen that before.
we collected a lollipop sponge. So if you saw the live yesterday, there were several of them, and we decided to collect one that was in the sand, and we decided to collect it with a slurp, which is kind of vacuum cleaner. And I was really surprised that it didn't go through at first. We had to actually dig a little bit around it to unroot it from the sand, and the root of that sponge was it was amazing.
Yeah,
I had never seen that structure before.
It was really sitting in
Yeah.
the sediment. Yeah.
So that's what I did this morning actually. I went to the sponge and studied it a bit more thoroughly, took some DNA samples, and I
And
think
it had juveniles as well, didn't it?
yes, it had little babies inside. It was reproducing. This is the lollipop sponges does direct development, so the little baby sponges develop in the mother directly without metamorphosis. So that was a very nice sample, an exciting sample for me, as well. So I should maybe here stress that sponges are animals. It's maybe not so obvious for people viewing us. Sponges are animals, although you see them still at the bottom of the ocean, not moving, but their larvae moves, their larvae swims around. Also, if you touch a sponge, they might move. Some species do change the size of their openings and so on, but it's very slow. But more than that, they share two characters that all animals have. They have a compound called collagen. Maybe you've heard of that protein. All animals produce a type of collagen, and sponges also do that. And also, sponges have sperm cells, and all animals have sperm cells. That is a characteristic that all animals share. So they are truly animals, although they are weird animals. I have to admit, not having organs or clear tissues, but animals nonetheless. And here we see that they really dominate these seamount habitats.
It's another soft coral.
Mhm. They can dominate these habitats to the point that we talk about sponge grounds, when the density of sponges is so high that there's many sponges per square meter, then we talk of a sponge ground. And here we are really not far from being a sponge ground, especially at Geodia and
It gets more
Roselidae.
rich now.
Yeah, a bit more
Uh,
dense.
You can see that it's increasing in animals. We have feather stars, we have soft corals, several feather stars in tiny place. Soft corals are increasing. We have lots of asbestopluma, the carnivorous sponges.
And what you might not see are all the little dots, like round little dots. All of these white dots or orange dots are also sponges. They're just smaller species. And you can see they're a bit everywhere as well.
Anemones are coming back.
Yeah. The pink dots are anemones.
Oh, shall we zoom in on that? It looks like a very dense polychaete wall, doesn't it?
That looks again like a wall of polychaetes. And there's a fish.
A very beautiful fish. Can we hold here for a sec, buddy?
Yeah, right.
Thank you.
Slow the ship to my warmer up.
Bridge RV, can we drop the speed to 0.1, please? 0.1.
What area you want to look at?
We want to look at this wall.
Just zooming in on the wall.
Okay.
Anyway.
One.
Thank you.
It reminds me of the wall of yesterday, you know, with
Yeah.
cracks below, and
Yeah.
it's not the same colors. It's not exactly the same sponge species. Okay. So Jenny is gonna zoom for us on that wall, because we saw a very similar wall, yeah, yesterday. And it looks that it's built a little bit the same way, with
3 meters away.
tube worms, Serpulidae. What did you call them? What did you say the common name was?
Bristleworms.
Bristleworms.
So what we have in the picture currently, to the far right, is an Ascidian, that is like the how do you say, this cylindrical shaped animal. We have a sort of a tree. It looks like a hanging tree shaped animal. That's a carnivorous sponge. We have some encrusting sponges.
A soft coral in light pink.
Okay.
Exactly.
Can I show something?
You want to zoom?
No,
Ah.
I don't need to zoom. I want to show. This is perfect. Look at this. You have the two different species of Geodia next to each other. So you have one species of Geodia with lots of little warts and lots of little holes on the left. That's Geodia parva. And on the right, you have a Geodia with only one hole. That's Geodia hanchelli.
And Hanchelli is the one that has other animals living on top.
No, it's the Geodia parva that prefers to have other animals growing on it. Geodia hanchelli usually, although here we
It's a little
see
exception, you know,
yeah.
I'd say.
it has some animals also growing on it, but it's nice to have them really next to each other. I like that.
Do you want an image without lasers?
I would, yeah, sure.
Okay, lasers off, please. Thank you.
So again, Geodia parva on the left with lots of little holes on top, and Geodia hanchelli on the right with just one single hole. And these are two different species.
We can log it.
I did.
Oh, perfect. Okay, because then we can include these images in the catalog
Absolutely, yeah.
for identification for
Yeah.
future cruises.
Yeah.
anyone who's visiting Arctic waters then actually has a chance
Mm-hmm.
to distinguish these species by footage, like by image and
Yeah,
video footage.
yeah. But it confirms that it's really a mix of the two species. I wasn't quite sure because I hadn't seen
Can move
Geodia
on.
handshelly.
Thank you.
Okay, let me go ahead first.
Yeah.
And get the ship.
Thank you, Patty.
Can move on with lasers on. Thank you.
Lights are on.
It's nice to see the wall full of bristleware. Ooh.
Oh, again,
Wow.
carnivorous sponges. Very happy here.
Yeah. What? That's yeah. There's a lollipop sponge, Paco.
There is?
Yes, there is. Look at it.
Oh yeah. Do you think you
Could
could?
we go around the corner and check that out?
I'm just trying to
Yeah.
see where we go.
Yeah.
Because I don't know what's at the other side.
Yeah, no, sure. Just looks so intriguing, you know?
I start to ship out.
A little peek.
Trying to be able to get them out from this corner.
Continue to move, yeah.
Yeah.
Bridge RV, can we continue the move now, please, at point 2?
Okay, so bring my head forward.
This looks really cool,
Yeah.
huh?
Mhm.
Okay.
But very steep. Very, very steep
Yeah,
still.
but I mean, we need these profiles for the currents
Okay.
to really,
Okay, Rob.
Mhm.
you know, the
Thank you.
currents just hit the wall and
That's fine, Rob, that's fine, that's fine.
bring the nutrients. Yeah, there we go.
It's bending around.
Massive.
There we
So I
go.
need him to be well around the
So we
corner
bring
for
our
the-
RV around to have a look. Look at this big anemone
One on a couple of
there in
days.
the distance. Really beautiful. Do we stop here?
Can we?
Are there possibilities to sample, or this is really too complicated? No.
Mm.
Okay.
Let's continue.
You have.
Yeah, we can if we can stay here and just continue to climb. So like
To climb in the corner
to have this still.
and
It,
Yeah.
climb.
Yeah.
Yeah, we can do that.
Okay.
Do you know, Jenny, if at the end of this transect there is a flattened area?
Yes.
There is, okay.
Yeah. So what we are doing is that we're climbing to look at the steep side of the seamount, which might have. Oh, sorry. There's a pycnogonid coming up. Talking about this area. Thank you. Do we see it here?
Oh wow!
And we
Yes.
see it-
Just coming in on the top right here.
That's it.
Okay.
Go yeah. Okay.
Pycnogonid is a sea spider, and Jenny and I are very fond of sea spiders.
Yeah.
That's
Sitting,
that's your cue. Maybe you can take my place,
Just be gentle.
and I'm gonna
Because it's tight.
take a pause, and then I will come and visit you a bit later,
anytime.
because then you can talk about sea spiders.
You still see it?
Yeah, it's there in the middle of
Yeah.
pink.
Yeah.
Yeah, on all position.
Thank you. You can do the zooming.
Yeah.
Okay. See you later.
This is
Thank you.
this is a beautiful sea spider.
You tell us when we
Yeah,
stop.
I think we can go.
No,
We're
I'm
ready.
waiting. I'm waiting for the pilot.
Yeah.
So rich. I think we can turn off the lasers. We have the size now. Thank you. Yeah.
No, we're still waiting for
Oh
the.
yeah, yeah. Oh good,
the-
oh good.
We're not.
I mean, there's lots to see. I mean,
Some anemones feeding.
yeah,
some-
soft corals.
That's a good spot for us.
It's amazing. Really good. Sure. This is beautiful too. I mean,
We will just wait until.
it's very rich suddenly.
Okay, try that. Yeah, tilt up a little bit, turn HD.
Wow.
Just need the umbilical sac properly, sac some stealth.
Oh, it's moving quite a lot. Little amphipod cruising by. There are two.
Yeah, you can see 2 sea spiders now on the right corner. And the one quite centered.
This is really beautiful.
I say what makes them so special.
Sorry?
Do you want to say, for example, what makes them so special?
Well, lots
the-
of things. They, for example, they carry all their organs in their legs. They are a group of animals where the male takes care of the babies, and the male is actually carrying them when they are born.
We can't quite see that. If this is a male or any of those two.
No, I wouldn't dare to tell.
No, but we saw some sea spiders with lots of juveniles on their back. Then we knew for sure.
Yeah.
But these two?
Look how rich it is. Look how much there's going on. This is incredible.
Stop the shifter.
Bridge RV, can we come to an all stop, please?
This could be Boreonymphon.
Yeah, the umbilical looks up here behind us, isn't it?
Yeah.
Stop. Yeah.
Copy that.
Mm.
Oh, this Zoom is doing a life on its own.
Okay.
Should be.
Sometimes it does. Do we want to? Yeah, let's go. Let's go. There will be more. That was very nice to appreciate them. Get them on the camera. Thank you.
Thanks a lot.
Okay. So we
We'll
going up the
continue.
corner.
Yes, up,
Yes, please.
Up, please.
Okay.
Can put the lasers back on.
Square up your camera there, please.
That's the fisheye we were talking about earlier. Shall we have it like this?
Yes.
So now we're climbing again. At this really beautiful cliff at 76 degrees north and a depth of 1,372 meters. And it's so beautiful and colorful. So what dominates this cliff are sponges, both filter feeding and carnivorous sponges, soft corals, some sea stars and anemones, feather stars, and of course the sea spiders, and then the little critters.
Sea stars.
Yeah, I wanted to talk about, that we have seen lots of little critters swimming by. We will focus on them later, right? Zoom in on them.
See down.
Sorry. really on this other camera that we have here in the ROV shack. We can see how far
Same move.
it goes
Bridge RV, can
down,
we continue the move, please?
and it really looks like a gorge, like we're just flying above the gorge, and there's nothing but depth. It's really, really cool. Right, thank you. A cozy live stream here today. Thank you. I mean, we are at 76 degrees north. Can't get away. A bit chilly. Let's see, I've just got an update from our office
Still can't see how
on the ship.
That's why the vessel stepped out because
Okay.
here in this corner. The moment the vessel
Thank you.
So we should be okay. So we'll fly over to this wall here.
There's another fish in the background.
Was it really good?
Almost lured me to believe it was a squid, you know, but then I realized that is a
Yeah,
fish.
that's good. So we're nice that you look at it. It's okay. Yes, it's coming all the time, so it's going okay. And come back to starboard.
So we have lots of large sponges again, and their faunal change. I mean, compared to what was on the edge of the cliff and what we have here now, there's much less because the currents aren't that strong, but still very diverse. See legs.
Something good, Rob?
Huh?
Has something good, is it?
Yeah. Good grading, yeah.
Okay. Let's watch that sonar.
Maybe
Watch
there
that
was a
sonar.
landslide.
It starts coming in the corner.
Some sediment drops.
Yeah, it
It's
looks.
very barren here.
Yeah,
I don't know.
yeah, it looks like it. There's a fish asleep. See it? Little rockling.
Oh, yes, sorry.
That is sheer vertical wall.
Yeah. And that's one of these other fishes again with the dot.
More white.
Mm-hmm.
One question on this one.
Another anemone. Pretty lonely.
Have you been to Spitsbergen?
Yes, I have.
What were you doing there?
I went to the University of Svalbard. So I studied in Bergen, at the University of Bergen, did my bachelor's in biology with a focus on marine
Bridge
biology.
RV, can we slow to 0.1, please?
And back in 2018, I had 30 ECTs that I could choose to do whatever
Thank
with,
you.
of courses. And I decided to do an exchange semester on Svalbard, on Spitsbergen. So I arrived there on the 5th of January 2018 in complete darkness, and didn't really know where I was because I couldn't see anything. I felt very disoriented. I couldn't point to where was north, or it was really a strange feeling. And I took 2 courses. Arctic marine biology and environmental management, and stayed there until the end of July. So I got to be there in the dark season, and I also experienced the midnight sun and the light season.
Both of the beauty, I assume.
Yes. Yeah, there was a very, very lovely time, and I met lots of lovely people there. I'm still friends with many of them. We really stayed in touch, and yeah, really lots of good friendships that have evolved there. How about you? Have you ever been?
Spitsbergen is the closest landmass from where we are here?
Yes, yes. It's about 215 kilometers away, so it's almost there. But only the southern tip of it. So Longyearbyen would be much further. Look at all the sea stars here now.
There are quite a few.
Yeah, actually,
I'm logging.
we are lugging
Sea star.
quite some of them. Have you ever been?
No, this one. I'd love to.
Yeah, if you get the chance, you should go. Oh, that was really stressed. Crustacean, I don't know. It's extremely beautiful. My personal opinion is that March is one of the most beautiful months because you have the sunlight, but you've still got the snow and the cold. I really enjoyed March.
Well, speaking about seasons, we don't know anything here about seasonal cycles. So, because there are so few samples, that any information we get also about reproductive stages is new. So it's interesting to see if animals that we collect have eggs or juveniles with them. Some we don't know if they're reproducing year round, or if they're dependent on the algae sinking down later in the cycle. Because of course there's no light down here, so that doesn't change. So the only changing factor is coming from the surface. Oops, what is this? Another feather star?
Yeah. On the sponge there.
Yes,
Yeah.
it was.
This is again another slide. Quite deep. Steep and deep. Sometimes they look like squids when you just see them come in, and then I realized, nope, that's decapods. And another sea star. But the soft corals are gone again. They're really patchy. It's like sometimes they're there and sometimes they're not. Another fish. It's almost, we only say fish. None of us are really fish experts, so it's hard to tell sometimes what kind of fish they are. Okay, now they're back. Now there are many soft corals. I haven't said a thing. Profile again. I'm interested in because we're almost at the end of T1. It was very short.
That's fine. We'll just continue.
Yeah, and it will
Straight away,
be
and then we'll have a
rather
steeper
steep.
climb.
It will be a rather steep
That will
climb
be interesting
for a
how
bit
this
more,
will change.
and then the area will flatten, so we can actually sit
Okay, yeah.
and have a look.
That sounds like a plan.
So we will always have like flat areas, some incline, flat area, and incline. It's pretty nice.
We're now at 1,350 meters.
It's like a mini cave. And sea stars again. Feather stars. Do you think this brown patch are speculumets?
Yes, I do.
Oh, is that a crinoid?
Yes. Oops, oops, I stopped.
Oh, let's just restart it, no worries. It might interfere with, let me do it. I would like to let the dry lab know that our OFOP stopped, and I currently have...
Sorry?
The OFOP stopped.
Shall we
Dry lab
a
ROV.
new one, or shall we go to
We need to do a new one.
Dry lab ROV. Yeah, the off-ops has stopped working.
Just restart. No, just restart.
We're restarting it.
go to the GPS logging?
Yeah.
To the logging one? And then go to the ship have you done the first two? The ship connection?
Yeah, I have. Let us do it in there and just name it.
We'll just name it B.
Yeah. Oh, sorry. So.
Okay, I'll start again.
Yeah. So what happened is, so we have a logging system here to log the fauna. It's called OFOP. It's pretty common to use for logging, and for some reason we had an error, and it stopped. So we will just presume the logging now, setting it up again. Takes a bit of time to set it back up. Just go to protocol, deployment window, just the first one. Yeah, and it's the same as you typed before. Right. Site X, not 2. Sorry.
And this is our 10th rough dive.
Yeah.
Station 4.
We need to go to deployment times. Videotapes is 2 hours. At the bottom.
So now.
Oh no, we might have overwritten the other one. I'm not sure. Let's. Okay. Just leave it. Fire, new protocol.
say yes, otherwise it won't let me proceed.
Yeah. Right. I was wrong. I think we have to do it again. Sorry. Let's just do it by manual. Yeah.
Okay.
It's a bit.
It's always a bit tedious to set up.
Yeah. Oh, good. I have it here. I'll just set it up. It's alright. There we
Okay,
go.
I can actually do it. I'll do it straight
Yeah,
from here.
I'll just keep an eye out for something that we haven't seen so far, and we'll just set up our off-up again. Yep. So if you look closely to your left, the left corner, there's a shrimp just hanging out on the rock, and he's gone. No.
It has to be number 13.
Yeah, you were in the wrong window. Sorry. This is all correct. It's all running correctly. We can just leave it run. It's all good. And then just proceed with the protocol, and it's all fine. So we've reached the first, or the end of our first transect, and we will proceed with the second transect straight away. This basically just means that we plan lines to fly in order for us to keep track on where we are and where we want to go. Because we could fly in any direction down here basically, but we need to know our direction before we start the dive, to not get lost and to be able to communicate to the ship where we are planning to head next. And we're also logging this information so we can trace this back afterwards. Something here.
Dry lab to Rough Shack.
Go ahead.
Did you manage to restart the off-up?
It's just recent.
Yes.
Yes, they did.
Thank you.
But where's the window now with the
One. Thank you. Okay.
videotape? No. The videotape window isn't here. Wait. Ron, sorry, what does your timer say on your video?
No, I'm just
Sorry?
How much is left?
No, I'm here on the time.
Oh, we started at 1953, so 21:53.
Okay,
Change.
would it be okay to do a tape change now so we
So we start
over?
again
Yeah.
with the tape because
Yeah.
Thank you. wouldn't do this now. Actually, I would just try.
Okay, stopping in 3, 2, 1. Stopped.
No, if we press stop recording when we stop recording, it should run. I think it should be fine. Let's just be good at logging the depths from time to time.
I entered and I also said that our system stopped.
Yeah.
Sorry, we haven't had that situation before.
Yeah. It's, things happen. Hey, Paco.
Hi.
It
You
just
know,
stopped.
offer a bit better. It just stopped, so we set it up again.
Yeah.
Just have a
But
seat.
we'll just wait until this the new tape is in. It looks fine.
Yeah.
But the other window isn't there. That's normally. Thank you.
I can take it.
No, don't do it now, while they're just changing it.
Okay. Which window is that you're missing?
Whenever you're ready.
Okay, thank you.
Recording. 3, 2, 1. Recording.
Okay, so I press this again.
Yes.
I have to.
Yeah, to start it. Yeah. Maybe we can write in the comments here, reopening of up,
Just give me a couple, 5
so that
minutes
we
to
remember.
work. Oh, sorry, start working. No, leave it. Sorry, leave it on port. I'll come around to that. But basically,
Up to 2 degrees to port.
22.
A bit more.
Yes.
So I just did that 2 minutes ago.
Okay.
Exactly the same.
And then it
Just check if that looks right to you now.
and you did the
Line there.
We did it exactly.
exactly
Yeah.
following
Just
the.
double check.
Okay.
Be on the safe side.
bring them right
We did
up.
it exactly went down the protocol. It's not possible to do it anymore. I guess once you're in that.
It's maybe here. No.
Could you please inform the dry lab that we just changed the videotape? Sorry, we missed
Dry
that.
lab RV.
Right, lad?
Yeah, the media has just been
It's here.
changed.
That's good.
That looks fine.
Good.
That
Thank
looks
you.
fine.
That looks like that wasn't running anymore.
That wasn't running.
That's
Okay.
what we had, and then
Okay. But here it looks
it
fine.
looks good now.
Yeah.
Okay.
Yeah. No, but I think we should be good.
I made a little remark here too.
Yeah.
Just what happened
Yeah.
between these two. You can stay if you like. I can be in the back seat for some time.
Sure.
No worries.
What do you want to do here?
No, it's alright. I'll drop in again. Just, I'll just bring the tape up and
Sure.
we'll
Yes.
pick up. Thank you. Do you need anything?
Yeah, I'll get myself a tea once you're back home.
Okay. Sure.
Later. That was a bit stressful, losing our screen. Oh.
So we are at 1,332 meters on the unnamed seamount.
More sponges, feather
Yes.
stars, anemones. Well, now that this logging system works again, I might as well enter.
So all the furry globular sponges you see here are Geodia, and there's 2 species. And the white vase-shaped sponges are of the family Rosetidae. There's 2 or 3 species in this area. It's very difficult to identify them without actually looking at their skeleton, so you need to sample to be able to tell the species. But they all belong to the same family of glass sponges. The way we identify sponges is by looking at their skeleton. Their skeleton is made of glass needles. They're the only animals on our planet to have a skeleton made of glass. So it's not carbonate calcium like corals or snails, but it's truly glass. And it's of various shapes and forms, and when we get a piece, we digest the tissue until only the glass remains, and then I look at the glass under the microscope. And by looking at the shape of the skeleton, then I can tell to which group it belongs or to which species it belongs. But for some of these sponges that are now quite well known, that we've sampled already several times, now we can identify them on the video, which is of course very practical for community analysis or just for telling you what we're seeing. Purple. Is that a sea star? Yes.
Well spotted. Very top corner. Has it got six arms?
It looks like, or maybe
No,
this
that
is
might
a
be the
part of
other
something
part.
else. Yeah.
No, no, left it. No, no.
There's a fish. There's quite a lot of fish on the seamount compared to our dive from yesterday. We were hoping to see more squids, but maybe they're a bit more shallow. The reason for that is that there was we've spotted whales, bottlenose whales, around the ship just before this live dive,
Yeah, thank you.
and these whales feed on squid, and they were feeding around the ship, so we were wondering if we would see a lot of squids, but maybe they're not that deep. We are fairly deep.
But these whales go quite deep. So why should they go deep if there's not their food? of
Maybe
choice?
that's true. That's true. Ah, that's one of yours, amphipods.
Yes.
How are we on the sampling side? Is it still complicated?
We're on the northern
but-
track now, so it might flatten
Yes.
out.
Okay. Ah, yes. There was a steep part of this mountain. So again, we are on the eastern side of the Mid-Atlantic Ridge, the Arctic Mid-Atlantic Ridge, called the Knipovich Ridge. And we're not too far away from Svalbard, about 200 kilometers from Svalbard. And we are 76 degrees north.
What is that?
Looks like a fish that was resting. Up to now, the community has not really changed so much these last 100 meters.
Camera. Right? I say camera. Yeah. Good.
we're still seeing pretty much the same type of community with occasional tube worm walls, Geodia grounds, and Roselidae grounds. And in the middle of that, of course, carnivorous sponges, sea stars, smaller sponges, anemones, soft corals. All in all, quite a rich community of species.
Bridge RV, can we come to an all stop there, please? Thank you.
Have you always been interested in sponges? Or when did it start?
No, I haven't always been interested in sponges. I've always been interested in nature. I was a bird watcher originally, but yeah, when I moved to Norway, that's when I started to get interested in marine science, and I met a sponge researcher, Hans Tore Rapp, who was a postdoc at the University of Bergen, and he was working on sponges. Especially, he was actually working on sponges from Greenland and the Greenland Sea. And he's the one who introduced me to the world of sponges. And he later became my supervisor, my PhD supervisor. So
Okay.
it's thanks to him that I started to work on sponges. And that was more than 15 years ago. So I'm still hooked. What about you? Who
That's
introduced
a
you?
very similar thing. I was always interested in marine specimens or the marine environment. And then while studying biology in Hamburg, I met Professor Angelika Brandt, and she got me totally hooked.
Where was that?
In Hamburg.
In Hamburg.
And then she became my PhD supervisor. And she was working on isopods. So it was kind of naturally that I fell into the crustacean group.
Mm-hmm.
Studied a different group of crustaceans, and she was specialized in.
Mm-hmm.
But she sent me to the right people.
And so you did your PhD in a museum.
Yes, and then yes, Natural History Collection of Hamburg,
Ah.
Museum Hamburg.
So here we have a sponge that we haven't seen so far.
Surprise! A sponge, really, Paco.
It's a bit too bright, but this is Asconema foliata. It's a glass
That's a-
sponge. We hadn't seen it so far. It's beautiful. Looks like a bouquet of flowers, or a cauliflower, maybe.
Bouquet is fine, I think. But Paco's having a very arty day today. He produced angel hair.
Yes,
in the lab.
angel hair from sponges. Yes, I did some nice pictures. I played with a stereo microscope this afternoon while processing the specimens, because I just find them so pretty, and I like to play with the light on sponges to make nice pictures. And that makes me think that we also, of course, on this expedition have an amazing photographer
Yes, so
that has been taking pictures of your animals, especially.
yeah, well, of all our animals.
Of all our animals.
Yeah,
Yes.
no, Solon Zankel, he's a genius. I mean, he does a lot of well, deep water, deep sea invertebrate footage. So samples that have been freshly collected when the colors haven't faded, and the ROV samples very gently. So some of the crustaceans, well, most still have all their antennae and long legs, so it's possible to see them in all their beauty, contrasting like a drag gear, where everything is meshed together.
Mhm. And he has an impressive equipment and setup up there in the lab. It's very interesting to watch him work,
Mhm,
actually.
mhm.
And he keeps those animals alive in tanks, and he has different setups to attach his camera to the aquarium and take
And we always
amazing
have,
pictures.
We always have to take care not getting any air to the animals when carrying
Yes.
them in, because
Very important.
air bubbles are forbidden.
Mm. So as soon as the robot arrives on deck, we rush with buckets of water, and
Yeah.
we try to bring those animals alive in those buckets, and then bring them to Solvin
And
for
registering at the same time, so
photography.
that it has our registration number connected to the photo. Because to be honest, after 100 pictures of very similar looking sponges, it's quite hard to place them. So they need to be registered first.
Absolutely, yes. We give numbers to every
Everything.
specimen, pretty much. This still makes me think quite a lot of what we saw yesterday on the Schultzbank seamount.
But it's far
Pretty
less
much at the
but it's far
same-
less colourful. I like colours,
Yes.
so
I'm expecting it might be colorful, a bit shallower.
no,
No?
I think like 500 meters shallower.
Yes. It's true, the geodias are not colorful, so you have these large potatoes everywhere, brownish potatoes. But we have today far more carnivorous sponges than yesterday. These little white tree-like or bush-like animals are carnivorous sponges. The ones you see on the crest right now. These are sponges. And there's a fish resting on the sponge.
I wonder what these sea stars eat. You see this
Oh, yeah.
two
Yeah.
ones? They have an interesting way of feeding. Putting their stomach forward and then holding their prey with their arms and then expelling some enzymes that digest their food but outside of their body, and then suck it all up
Oh, yeah.
and walk on. I just wonder what they're eating down there.
Yeah.
Normally little like shells,
Gently, because I don't want to trust the
about
carnivore
shells,
watch.
the inside of a
Watch
shell.
my gently.
Mhm.
So every meter you can take a meter, don't you? Yeah.
Maybe they're hungry.
We saw one a couple of years ago that was that we found on a stocked crinoid, or was it a sea pen? I don't remember. We took a nice picture, and it's one of the few sea stars we saw actually eating. But they're voracious. I mean, they eat a lot of things here. Oh wow! This is finally flattening out. We're at 1,300 meters, and we seem to have reached some kind of flattened area.
Would it be
Plateau?
Would it be possible to sit down with the rough? Flat?
Yeah, I'd have to fly in a few meters.
Yes, for sure.
Sure.
Sure. But whenever you see a
Yeah, sure.
flat, non-spongy spot,
Yeah.
So since we started, and that was about 40 minutes ago, we've been ascending non-stop this very steep cliff, and it was impossible to sample anything. It was too complicated with the ROV, remote operated vehicle. But here finally it seems we'll be able to settle and take our time to zoom in and see the sponges and maybe even sample.
but we can also fly on whatever you prefer. Do you see a sponge that you want?
Right now,
No.
no.
Then let's fly on. Sorry, I was thinking of the coffee mug, of the sediment, but we might as well do this in one go.
Yes,
So whenever you see a sponge, then we'll.
that's a good idea.
Yeah.
Yes, that's a good idea. Next time we stop, let's do the coffee mug and a sponge. But actually, this could be a one. This one here. So I've just spotted in front of us a little pompon sponge, little bushy-like sponge, white.
Our colleague Sergi is very interested in those.
Yes,
So
he's interested in doing population
Genetics,
studies,
yeah.
population genetics.
It's this one here in front of me, isn't it?
I think it's next to this one. It's this one. So it's this one.
but also they will start to grow more and more, don't you think? While they're going shallow.
It's giving a bit of wear off.
Probably.
So let's
There you go.
Because here it could be nice now to use the mug, but
now might well be a bit muddy.
let's see what it looks like when we sample the sponge. Maybe you'll get an idea of the terrain.
How do you want to sample it?
With the slurp.
Okay.
You have the papers, I think. Yes.
Do.
This will be our first sample,
Cast one.
and for this we'll probably have to remove the fisheye.
Where it's at. Yeah, that's the start, isn't it?
Yeah, that's what's stopped. What tank number, please?
So it will be slurp 1, compartment 1.
Okay.
You have it.
Yes, from 16.
Yeah. You bring it in wrong.
Sorry?
I'm bringing it.
Okay, I'll do this slow.
We'll just sit here.
Yeah.
Okay, that's all it.
Do you want a close up?
I will now just have a close-up of the sponge before we sample it, so that we have the scale. Again, the lasers are spaced 20 centimeters apart. So that sponge is about 20 centimeters, at least the bottom part, which is all I need.
You can see the little whelk, the snail in the front,
Oh yes,
the white one.
one at the bottom?
Yeah.
Yeah.
Buccinida. And you can also see the other little snails, they're more flat
Yeah.
all around. Do you mind if we zoom out a little bit?
No,
Just zoom
absolutely.
out and just stay and go to the left a little bit. I want to see this red one here, just sitting there. And back in again, please.
Yes, that's an amphipod.
I know. Well, they're two amphipods, actually.
Are you doing
I would-
the high flow?
Whatever. Yeah.
Ooh,
You see
I would love to sample that when we're sitting there. Can I go in the same slope?
Absolutely. So we've spotted a really nice.
We'll come back
And
there.
also really nice amphipod, maybe that we could
That
slurp
we
later
have
on.
not sampled on. You see all this the red
Oh yeah, there's
eyed
another
ones,
one.
so there
Wow.
are quite a few, and there's one sitting in the front here and right here.
Yeah.
So,
Amphipods.
well, we can do your sponge first,
Yeah, absolutely. We can do the sponge
and they
first.
go in the same slope. Yeah.
Yeah, no problem. This will be cast 1.
Oh, sorry, I'm trying to steal you. Just going to your.
Yeah. No problem. I can come back here.
Yes, please.
We are ready to sample.
Lasers off. I don't mind.
Oh, it's okay.
You just zoom back out a bit. So
Sure.
you can see her coming in here.
Absolutely.
Yeah, there you go.
Now you see the slurp. It's like a vacuum that will suck the sponge.
So we're good to go there, guys.
And we just need a part of it, so
Yeah.
if we don't get all of it, it's perfectly okay. Yeah, this is good. Very, very good.
In you go.
Very
Right,
clean
yeah.
sampling.
Okay.
Thank you.
pods then you want? Are
So they are
Just to
you
the
see,
left.
they are
Can you
just to the left here.
Can you move a little bit?
Yeah.
Same tank
So-
or?
yeah, same tank.
There are two, aren't there?
So there was
there
a second
two large
one,
ones. So
here.
wherever is easiest to reach, I'd like to sample those, please.
So right.
Should we do? How about it?
Go around.
Really?
Yeah. Good. And one to the left.
And there's
And the
an,
next one.
yes,
Maybe show them on the pics.
yes.
Can you see them?
Here, there's another one.
Look.
So the bright spot we see are their little eyes. There we go.
Thanks a lot.
Thank you very much. Oh, I'm sorry. Is that one too close?
We might be able to get it.
So while the roof is stable,
Yeah.
Paco has discovered another
same
little
tank?
sponge. Same
Yes.
tank, all the same. All slope one. This one or this one?
Yes. This one, perfectly. Yes.
No, a little bit to the left again.
Right, plus.
Yeah.
Perfect. Oh! It's well attached maybe to the big rock. And if that's the case, then
Slide it down now.
Yes.
Yeah. It's stuck on the stone. There you go.
Try it again.
Try again.
perfect.
Snap.
Oh,
Nope.
Oh.
The stone is
You can
too
just
big.
scrape it off, don't put it on yet. Just scrape it off.
so we are scraping the sponge off because it's attached to a large rock. This is a technique. Yes, we got it. Thank you very much.
Okay.
What's that one called?
I don't know. I've called it round white sponge.
You're great. If the expert on sponges calls it round one. Okay.
Yeah.
Thanks a lot, guys. It was a very successful sampling trip.
Yes.
Highly
Okay.
appreciate it.
We move on.
Yes, please. We took down 2 amphipods as well in there.
Yes.
I don't have to take a muck here because we've got enough sediment sucked in with it.
That's
If,
true.
if there were some
That's true.
tenacious commissions, we'll
Mm.
get them. We'll sieve them through. Now what's this sitting here? A shrimp?
Yeah. Yes.
Here, I'm going to have to get the vessel moving now.
Can you move to the left a little bit?
No,
For the feather start
keep
to
it all
come
in
in
more, so
focus.
we're getting moving.
15.
It's nicer to see.
015, yeah, 0.2 for the moment.
Bridge RV, can we get a vessel move please? 015 continuous please, at 0.1. Thank you.
Tight, is it?
Huh?
Tight, is it?
Not really.
So here we have a different type of landscape. It's still large Rosellidae glass sponges. I see lollipop sponges as well. It's much. Oh, there's a
big fish.
large fish
Just
here.
wave towards you.
Yeah.
I
Can you
know
tell
you're
me
distracted
when-
by the fish, but can you look at the red part on the sponge, please?
We're getting.
It's an anemone. Don't worry. It's a
So we're very privileged here in this expedition that scientists can actually play with the camera. So I have two joysticks in front of me. One for
one is mine. This is zoom.
that is true. One for going left or right, and the other one is a zoom, which is
Yeah, and down there.
yeah,
More amphipods.
amphipods swimming. And it's very convenient, of course, when we want to look at things in detail without disturbing the ROV pilot. They have their own cameras as well, of course, which enables them to see all around the ROV. So as you can see, as soon as the rocks disappear or the hard bottom disappears, a lot of sponges disappear because this is very sandy, this is very soft. The sponges don't like it so much unless they can root themselves in the sand, and that is the case of the lollipop sponge. It can, we now know because we found one yesterday. We dug it out of the sediment and we found that they actually produce really strong roots.
Like one long root.
Yeah.
Not like many branches, but little branches from one,
Yeah,
like a stem root.
yeah. Super impressive.
No Birdle stars.
No. And we've had also dives where there were jellyfish everywhere. Here there's none. But it was also maybe deeper. When we were at Loki's castle, we had these red jellyfish.
It's an Ascidian.
Sea cucumber? No.
Soft coral.
Ascidian, yes. So these transparent little bags, floating bags at the bottom of the ocean are ascidians. These are animals that have two holes, one for in-current water, one for ex-current water, and they filter. They're also filter feeders. They feed in the water column just like sponges, except that ascidians belong to a totally different group, which is actually quite evolutionarily connected to us. They are called chordates. So during their development, they have some common steps of development that they share with us, believe it or not, because of course they look so differently, so different from vertebrates. We're swapping regularly because, well, Temperature, yes, is one of the reasons. It's a bit chilly here in the rough shack.
It is. I saw you just collected a sample.
We did,
Congratulations.
and amphipods, 2 amphipods
Oh,
in the same slurp.
were we slurping? Oh, nice.
Yes. Yeah, maybe I can give this back to you actually.
So,
So it was cast 1 for all of this. Maybe I should.
so we slurped in the bio box? Oh. Okay. That would be new.
Wrong paper.
No worries.
Let me rewrite that.
Sorry. It's getting late.
It's getting late, yes. So our night shift will happen at 2 in the morning.
Our shift change.
Our shift change.
Yeah. Yeah.
So we still have a few hours to go.
Oh, there was a rocking asleep. Yeah, depending on where you are in the world.
Yes. So there's nothing in the bio box 1.
It's all feet now, this is no sharpness.
So I've collected actually one of these white sponges because I have no idea what they are, and it was just on the side.
Yeah.
Alright, yes, Jeff.
But yeah, they seem to be quite abundant.
We have push cores and mugs, and
Yes,
as always,
yes.
do we have one mug?
I believe we have 2.
Mm-hmm.
But maybe next time we sample a sponge, we can absolutely take a mug sample.
Mm-hmm.
Bring my heading to 000.
It's also nice to show the people watching how deep sea biologists sample the deep sea with the ROV, all the tools that we have.
I think so too.
And I can also remind you that once this live is done, you can rewatch it. Actually, it will stay. It will stay online
Mm.
if you've missed part of it. So the rocks have disappeared, and with the
Yeah.
rocks, most of the sponges,
I can see
and the
that.
other animals that need to
Is that a
settle.
sea star?
It
Is
is.
it right?
Yeah. Do you need to sample sea stars?
No.
Not particularly, unless it's something that you've never seen before.
Yeah, no, it's
It's so different now.
really, yeah. It's interesting because I would have expected more rock,
Mm-hmm.
but then again, we saw these sedimentary. We saw that sediments had been falling down from the top,
Yeah.
and it was quite a sedimented slope for being that steep, sort of. Mm.
So what you have to realize is when we look on the maps, we have no way of knowing what the terrain is, if it's going to be hard or soft.
It's
we
hard
can-
to guess. Yeah.
We can guess. If we see a ridge, we can guess it's a bit hard. But quite often we're surprised.
Yeah. Like now.
Yes. So we would need other types of data.
Are those the sponges with T? The tentorium?
They could be, yes.
Okay.
They could be. I'm not 100% sure. They could also be,
Oh, there was a whelk.
yeah. Yeah, they could also be another genus of sponge called Polymastia. Could we subsample that large sponge?
Okay. Slurping or picking?
Picking. So I've spotted this sponge. I don't think I know what it is. I think it's a glass sponge by the fragile aspect, but
We're sitting down.
I'm very curious to see if we can grab a sample to bring in the lab. So I'm going to first zoom as soon as we are settled on the ground. Maybe this could be a good place for also a mug
I agree.
sample, because we have a lot of space around us.
How about we take the mug first and place this on top?
So this is going to be very fragile, as you can
Okay.
see.
Okay.
I would put them in different bio boxes.
We can do that for sure.
Look at the details. There seems to be a margin all around it. So I will definitely send a picture of this specimen to our world specialist, Celso Domingos. He's a Brazilian researcher, expert on glass sponges. He might be interested in this. We can sample. I think it's going to be very soft and very fragile.
Is it very light?
Yes, it's going to be very light.
And you just want a small sample?
Yes, I can have a small sample. I don't need the whole specimen.
So just grab a little bit.
If possible, yes. If everything comes, it's okay. We can place it in a bio box.
Port side, please, because then we can take the mug out afterwards.
Always not.
So you're going to see another technique using the automated arm.
The manipulator, and it's so strong. It's incredible. It's like I don't know.
It can bend metal.
Yeah,
We've
like
seen that, right?
easily,
Before.
But then
Mhm.
again, you can sample so specifically
Yeah.
and careful with it.
Yes.
So
So Andrea is currently controlling the arm. So it needs to be a different person from the ROV pilot, because these are two different tasks. You cannot do both things at the same time.
wow, it's super rigid.
It's tougher than I thought.
It's
So-
not fragile at all.
No.
So we can suck that up. She can pick it, we can suck it up then. And you put that in the box, it's going to float away.
Yeah,
Okay.
you're right.
Okay.
You're right.
Let's do that. You think the sucker will be big enough? No.
If she plucks it,
Aha.
And then
And
we
then
suck
suck
it.
it.
Yeah.
Okay. Yeah, that works.
You're going to snap it off and put it slow away. You get off a bit longer.
Wow, I would never have expected this.
It's like rubbery in a way.
wow.
Oh, we have a small piece.
This
That's
is
enough.
Yes, that's enough.
wow.
With a small piece like
Stand later.
this, we can have DNA. We can have a piece of the skeleton as well. And the sponge is fine. It will heal and regenerate.
so what tank are we putting that in?
So,
Sorry,
slurp
Patty.
2,
Yeah.
compartment 2.
2, please.
Yeah, one sec. Okay.
I will write a small piece of
glass sponge.
glass sponge and fern, maybe.
Yes, yes, you can write that.
Because in case they want to slurp more, then
Yeah, yeah. I think the
Oh,
piece
yeah.
is about 10 centimeters long. So you have to imagine that at the back of the robot there are several compartments connected to the slurp, and we have actually 5 compartments, different compartments separated, so we can sample different things that will not mix. So we are putting this sample in compartment 2.
I have a photo of this.
Can you zoom in a little, guys?
This is a technique I have not seen often.
That's up. That's up. No, like, don't put it out because your jaws will crack the thing. So just come to the side.
This is cool.
this is really cool. It's a really nice
Have you
technique.
seen this before?
No, it's a new
It's all right.
new thing.
Yeah.
Hold on. No, stay there. You just come around.
I destroyed it. There we go.
Wow.
This is really nice.
Touch. Okay, I'm gonna put her on. So don't let it go, Andrea.
No.
Okay, it's on, let it go.
Amazing.
Oh, wow.
We have
We have a delivery.
Success.
We have a delivery.
Let's go, Mike.
So we have a little camera in the compartments behind the ROV where we see if the sample has arrived safely, and it has.
No, just there because you're going to take it. above.
Beautiful.
Just wait there. Get Rob out the way.
And now, in addition to this sponge sample, we are very interested in what kind of sediment we have here, and we would very much like to have a sample of this sediment, and we are going to do that by using our coffee mug, as we like to call it.
Mm-hmm.
okay.
You will see it very briefly, or maybe you've seen it in the previous live stream of our dives. And today we're diving with 2 of them actually in our port side drawer.
You can see, you already see the handles there. So again, Andrea is controlling the manipulator, catching the handle of these mugs, of one of these mugs. I am directing the camera towards the mug to help Andrea have a feeling for the focus. I think that's the most challenging part while handling things in the deep sea, is to have an idea of how things are positioned in front of you. It's very complicated.
And in the meantime, I am just writing down some notes about what we are actually doing.
Of course, yeah, anytime we take a sample, we write the coordinates, of course, the depth, and we give it a number. Number for a sampling.
1280 meters.
Yeah.
Yes. Or actually, zoom in on the sediment, please.
Yes?
Just right before we sample.
Yeah.
What are these things?
We've had these before. I think I think they're foraminifera, but I'm not entirely
There's
sure.
an amphipod crawling. Anyway,
and-
anywhere, please. We can zoom back out. Sorry, I just wanted to confirm.
Yeah, no, but it's good to have also a good close-up, you're right.
Yeah. And we might want to take quite a... let's see how the sediment goes into the net.
Mm.
Let's see what kind of sediments
Yeah.
we're dealing with. Yes.
Oh, it's very soft.
Awesome. Thank
Yeah.
you.
So if you can shake it a little bit to try to get the fine dust out. Oops, sorry.
No worries. I was
Yeah, you're right.
interfering in your territory.
No, but it was a good thing because I had forgotten to follow the arm.
This looks very good.
Yes. Oops, sorry.
Hoppala.
Wrong.
Awesome.
So that's a nice chunk of
Yeah, it's
mud.
really good.
Good. So this mud will be sorted on deck and we will look for macrofauna, which are animals usually bigger than 1 millimeter. And it can be crustaceans, it can be sometimes foraminifera, mollusks, etc.
This looks perfect.
Yeah, perfectly handled.
Yeah, really well. Thank you.
Thank you.
Cool.
Okay, that's it in Port Royal, yeah?
That
Yes.
is. That's it. I'm good to go when you are.
I'm good to go.
Your mark, please.
There's another tumbling
No, we're flat now around the top.
sponge, I think.
Yeah.
You think so?
Okay.
It's very strange. There's a tumbling. We're not quite sure if it's a sponge. We've seen two yesterday, right? And we were
Yeah.
very surprised to see tumbling sponges in the deep sea. But yes, I think you're right. This is a tumbling sponge as well.
Okay.
I mean, currents can be quite important in the deep sea. Do not think that everything is quiet and very slow in the deep sea. There can be waves, very violent waves and currents.
What's that?
I get the ship moving.
What
He's
is
still
that?
moving, isn't he? The ship?
No, it shouldn't be.
Ah, it's just rock.
Yes.
Yeah, it's a rock.
Sometimes
it-
the shadows are strange and
Yeah, it did
It's
have
leave it
an
like this.
unusual shape.
can live in a bit zoomed actually.
Zoomed in.
Just a bit. Yeah.
This-
Yeah, I think that's nice. So we see a bit more of what's going on. Should I log
Geographic, can we continue the move, please?
It's all Rosselidae, isn't it?
Yes. Yeah, except the one we sampled, I think
Yeah.
looks a bit different.
Yeah, I think so.
And it's not something we had sampled before, so I'm very curious of that sample.
Oh, beautiful.
Mmm.
Just getting a couple of degrees support in my head.
And look, it's there again, this very
Mm.
fine veil, but actually quite strong.
RV.
Very firm, yeah.
Yeah, can you continue the move, please?
Can continue it.
Yeah.
I haven't stopped.
Okay.
So now we're seeing a few rocks here and there,
And boom.
and boom, little oases of sponges. We are at 1,277 meters at the unnamed seamount on the eastern side of the Arctic Mid-Atlantic Ridge, 76 degrees north.
And we believe that no humans have been down here before, ever. And that these are the very first recordings and sightings of life at this particular environment in the Arctic deep sea. Now look at him
Oh,
or her.
Just give me another cup. Take 4 drops.
Hey.
Bring in that gently your tie drop. Pull it.
I love them. These feather stars.
looks like a different species of soft coral.
I believe you're. Yeah, I think so too.
They're short.
Could be small ones though, but
It could be, but they're all the same size. That's why I'm.
different species.
Thank you.
Is that a fish?
I believe it's a fish. A lot of fish on this mountain. Oh, what are these balls?
Oh my!
Can we stop? Looks like a sponge cemetery, or what is that?
That's
Hey, stop the ship. Stop the ship.
Great job, Ray. Can we stop the move, please? There you go.
so. I will write eel pout type of fish
Yes.
because I don't know.
Whelk.
Well, okay. Now there's lots of things here. What do you think? Geodes?
No. No. It looks. I think it's the same thing that was tumbling along. These are. I'm not sure. I really think we should sample one of these things because I would like to know if these are dead sponges or living sponges. These are definitely not algae because we're far too deep to have algae. I've never seen anything like it, to be honest.
Do we reach them from where we sit?
Don't think so.
No. Can we scooch?
Yeah, we can. We start them up.
Do you think slurping?
How else are you going to get them?
Do we bring the
Could we
the scoop?
Scoop or
Into a bio box then? Yeah, we
Yeah.
can try that, yeah.
Let's scoop.
yeah.
Okay.
I'm
so
afraid to
strange.
slurp because
Yeah,
it will.
sure. No, so
Because when it was tumbling along, it didn't seem very. Look at that.
So
And they're all accumulated here.
we promised you to show you all the gear we brought down here. So we are really, we're good at this. We've had the slurp, we've had the manipulator and the slurp. Now we're scooping. We've used the mug.
Scoop.
This is great.
Okay.
We show the entire variety of our gear basically, which is extremely important to identify and catalog what we have of life in this environment.
Oh, look at that.
Of course,
Moved.
just when the whelk was in the image.
It's
you had
it's not
to-
me. No, it's the ROV that repositions itself.
Right.
So this fish is about 30 centimeters long,
Yeah.
something like that. And we have these. Honestly, I don't know. Are these carcasses of sponges? I don't know.
I would sample this.
This one?
Yeah, I think so.
Let's try.
Or this, depending on what we can
Yeah.
reach better.
okay.
Yeah. Should we put it in a bio box? We try?
Yeah, let's see.
Okay, 4 box for you there. You need to get them
Port box?
in. Yeah.
Yeah. Sure thing. Anything, whatever.
Okay, go.
except for the one with the mug.
Yeah.
Yeah.
So 3 or 4, whatever.
Yes.
Right.
Absolutely.
All right. We.
I think the fish is in for a surprise.
You need a move, buddy.
Okay. Maybe I should zoom out to see, to show you a little bit what's going on. So this is the scoop.
This one's.
Enough there on it, up to me, it's good. Enough, I think we should. It's not up by a partner.
So for those joining us now, we're at 1,276 meters trying to sample these fluffy balls that we have no idea what they are. We've just, they've been accumulating behind this small...
Zoom back up.
You.
Hey.
Would you like me to zoom in?
That's fine.
That's
Yeah.
fine.
Oi. Interesting.
Are these?
Good more.
Okay.
This is very interesting. Just follow it.
Yeah.
Okay. There's lots going on in this sample. I will write a little note that this is very sedimentary,
Yeah.
this biobox, because we have already taken a mug. This will be the...
Yes. I wonder if these could be sponge spicules of dead sponges that are rolling and making those balls. Just like, you know, when algae also die, sometimes they make those balls on the coast.
Whichever one you get then.
But I mean, this would be very interesting.
That one makes sense. Whichever.
I don't believe this has been described in the scientific literature. I may be wrong, but definitely worth investigating.
Yeah, for sure. I mean, we basically do not know anything about these types of biological...
Get that one off.
Yeah, that is perfect.
So that is kind of four.
Yeah.
Thank you, Rob. Rob is currently controlling the arm of our ROV, and therefore the scoop.
You know what will be interesting?
And the fish is still there.
Oh my god. It will be interesting to actually sieve the water and see whether there are any critters that live inside of this
These
sponge
structures,
fluff.
yeah, absolutely.
I mean, we have had quite some training on this sieving, so I will actually call this.
It's clearly not alive, but it's a structure maybe that's made of either dead. So, well, all in all, an interesting sample.
Absolutely. Now, whilst we're still sitting, I would really like to get a zoom on the crinoid just because it's pretty.
Okay.
And after that, we can continue.
Oh, there's a little anemone as well. And we have our soft
and the
coral.
Isn't that just a beautiful picture? I like it. We can have the lasers off there, please, for a sec. What kind of sponge is this?
Oh.
Thanks.
This looks like a Roselidae. It's a little bit strange shape, but I think it's a Roselidae for sure. It's one of the common ones we've been seeing here and there. There's 3 species in the area, but it's nearly impossible to distinguish them without sampling and looking at their skeleton. But it's definitely a glass sponge.
I think it's cool. We have a glass sponge, a feather star, a whelk, an ascidian, an anemone, and a soft coral, all in one picture.
Mm-hmm.
And this is no more than...
And the fluffy balls.
And the fluffy balls. And your some other sponge species.
Probably Tentorium, but I'm not exactly sure.
And we're looking at like what, 50 centimeters of seafloor here?
Yeah.
It's insane.
Yeah.
Thank you. That was perfect.
On we go.
We can go.
Yeah, that was beautiful.
Look at that. It is intriguing.
The rock is like a barrier.
Yeah.
And it just piles up. Very interesting.
I mean, one of our aims here is to understand these communities, and seeing these little balls, I think, is interesting. We've seen spicule mats, we've been seeing tube worm reefs, we've been seeing bamboo coral reefs, sponge grounds, so many different types of habitats, and every time we understand them a little bit more by sampling species,
350.
identifying them.
Bridge RV, can we get a vessel move, please? 350 at 0.1, please. Thank you.
Just checking. That was a 10-4.
Yes.
Ribbebanet in Norwegian.
I didn't know that.
I kind of like it because it's a very descriptive name. It has... Yeah, there they are again!
Always protected by a rock.
I will write it down. Round sponge fluff.
Yeah. Look like little fluffy. It's very intriguing.
This is quite a habitat change, huh?
It is.
From what we had in the beginning.
Which was intense.
Very much, yeah. There was lots going on.
Yeah. So we're now following a second transect, different from the first one. That should lead us to a ridge of some sort. Or this is already the ridge.
Yeah, it's at least we climbed to the top of the, or not to the top of the seamount, but to the flat part of it.
I mean, one of the questions I had in my mind is, will we find a sponge ground as well on the summit, which we have not reached yet? Maybe this is a question we'll be able to answer near the end of this dive. One thing maybe to keep in mind is that this unnamed seamount has a summit that's not as shallow as the Schultzbank seamount. Right?
Yeah.
So it is a different setup.
Definitely. It is. And we never know whether it's sedimented or rather rocky. This now is sedimented, but what is good here is that we still have some coverage of rocks that animals can cling to. but this is very poor, I'd say, in comparison to the slope. But the slope, I think the most interesting about this is actually that, let's say you would start the dive here and you would have a completely different idea of this seamount and its ecosystem. However, we started the dive on the slope and we know what kind of animals, species are here and that their larvae are around, but they won't settle up here because the currents are not strong enough and the substrate is just not sufficient to settle.
It's been 1.2 Rob.
Bridge RV, can we go to 0.2 please?
I agree.
They're all here. It's just that they need specific environments to settle.
The GPS.
One thing I can say as well is that although the sand
No, leave it off.
looks empty, it is actually full of life.
Yeah. Look at this whelk on the
We
rock.
just don't see it.
Isn't it beautiful?
There's
It's alright. Crawling up.
this is why we do mug sampling or push cores. That's another technique we haven't done so far, is to actually be able to look inside this sand and find smaller animals, which are usually very abundant. So this is not a desert at all.
We sorted one mug of sediment and we found I don't know how many polychaetes, like bristleworms, worms that live in tubes, for example. But not necessarily all of them. Some do not live in tubes, some just bury in the sediment. And sediments are always very rich of bristleworms. You always find bristleworms. If you don't find them, it's really odd. Usually we always have them. So what is the water temperature here? So like, okay, minus 0.9, minus 0.3.
Yeah.
Oh, sorry, I'm yawning. That's beautiful.
Alright, just.
Hi.
Hi. Hello. Hi.
You're on the bridge.
Yes, we are. We are on. I can actually see what's ahead of us. Is it true that the ROV is so far off the track?
Yeah, because we'd come around the corner. Otherwise we wouldn't have enough ability. Remember, you wanted to come up in the corner?
Sorry?
Remember, you wanted to come up on the corner?
Yes.
Yeah, we need to bring the ship around to do a loop, otherwise our ability will get stuck coming up.
Okay.
Or looping back up onto it.
Yeah, no worries.
Yeah, well, just we could do that.
Yeah, no worries. It's just sometimes. Yeah, no, never mind. All good.
You speak about the pine tree, Rob? Hello, don't actually don't.
Which way we need to go with him?
Fine answer.
Final answer, yeah. Final answer. I'm just trying to think in flight center.
I was interested because do you see the shadow here?
Yeah.
And so if we
seems that here it's gonna get like there's. Is it a canyon or is it a ridge? It's
No.
difficult to know.
Right?
Mm.
So now we're a bit, we dive like this and then we go back in. So this is all sediment, but it doesn't have to be here.
I mean, you're witnessing exploration. That's what exploration is all about. Not knowing exactly what we're going to find. And the cliff was already totally unexpected, and the abundance of sponges and life on it. And now we have this kind of flat plain in the mountain.
Mm-mm. If this was on land, you would have moss
Yeah, or
and lichens. And
Grassland.
Grassland area.
Make it any easier to do it.
Go ahead. Maybe come 3 meters.
there's a few lollipop sponges.
Really?
Maybe, I think. No, it's something else.
I was just about to log Styloquadrilla.
Mm-hmm.
This root thing I will never forget this. How they have roots and are really rooted into the sediment. That amazed me.
Yeah, me too. I think this is what we sampled earlier.
Okay.
It seems to be quite common. So I'm eager to try to put a name on it.
Ready? 10.
I mean, this would also be a perfect environment for sea cucumbers, don't you think?
Exactly. Yes, I do.
Why are
think.
they not there?
I don't know. I really want.
Or brittle stars, for that matter.
Yeah, but we're too far away. We need to do some zooming.
Maybe.
But
maybe.
we should maybe just sit. Can we sit in front of that stone that's coming up right in the center?
Okay. Stop the ship.
Thank you.
Bridge RV, can we come to an all stop there, please?
We need to look at this. This
Yeah.
is. This
Yeah,
is-
there's a couple of interesting things there.
Yeah, and I need to look at the sediment. Something. There's something.
Isn't there a rock, a fish
A rockling. Yeah. Yeah.
there?
I loved it. This is pretty.
Yeah.
Very pretty. I hope he stays. Are we sitting?
Yeah, go for it.
Yeah.
Oh wow. I was expecting some sediment plume and then there was just nothing. This
Sorry,
is really
I'm
nice.
trying to get the best
Yeah.
frame. Look at this.
Let's just hold this for some time.
Mm-hmm.
We are at 1269 meters depth. This is a very nice still.
We have feather stars sitting on glass sponges. We have soft coral on the right. The pompon sponge, the white sponge on the right as well, with an ascidian.
There's nothing else, was there?
And there's another hairy sponge covered by all this. And I would think it's a geodia probably, but it's difficult to be sure from here. And then we have the fish.
And in the background we have some bristleworms, some sabulid bristleworms. May I have some?
Absolutely.
I want
Be my
to.
guest.
You know what? Let's just sit and wait for a couple of more seconds. Because in addition to that, we are creating video files and video footage. We also want some still images, right? To show to the public, to you, to whoever is interested. We want to use this in our publications, so it's nice to just sit.
I guess you will want to watch what's going on there, maybe.
Yes, and a bit around in sediments. Okay. Hello.
But if they finish this, relocate, and just start going straight away.
I like how the fish is using its fin to hang out.
Just get you to stick up on the board there. Take away the old one.
Let's see if we can. So we are looking at bristleworm tubes. There's a little gastropod in front of the stone in the center, or a snail, if you like. It's a very bright snail because our lights are just shining right at it. And then we have an amphipod moving to the left of the very bright little snail.
Oh yeah, I see it now. Just below the snail. You can see its shiny eyes. And the second one.
And the third one in red, just crawling here.
Oh wow.
Also below this. There are lots of
Oh yeah,
amphipods.
they're everywhere. Now I see them.
They're everywhere. We just have to look closely.
Sorry to disturb.
No worries.
Just updated for Transect 3.
Oh, perfect.
So we can do that right after this one.
Okay.
Yeah.
Thank you.
Okay.
But you know, I do not see any brittle stars.
No. So that answers our question as well. No blue stars. I wonder if these are remnants of lollipop sponges, you know, when they die. And I think the stalk may remain in the sand.
I'm not so sure.
Or tube worms, maybe.
I do think it's tablets.
Okay.
I have to say, I do really think so.
I think you're right, because here the angle of them makes me think more of a worm.
But it's good we had a peek because this is very dominant of the soft bottom fauna here. Also the white gastropods, now I see them everywhere. You see? They're everywhere.
Yeah.
It's just that we didn't see them before because they're just white dots on the sediment.
Yeah.
Okay. I think we can continue.
We're
Okay.
good to go.
Right.
Thank you. We can put the lasers back on and I think they're off. Thank you. Now they're everywhere, these little gastropods. Look, suddenly we see them. All these white little dots that you see are snails. That's cool.
Mushrooms?
wherever you want to go. Okay.
Is that?
No. Oh, fine. Off you go.
Those are the ones we sampled, maybe.
Yeah, maybe. Maybe.
So you know I
Hmm.
They could be. Yes. They look so different from.
Hmm.
Quite strange.
You guys want to jump out?
Yeah, I'll get you out there.
Oh, there's that fish again.
Mm-hmm.
Yeah. We're just going to change over here, guys, okay?
Sure. We're changing the ROV pilot and welcoming Will.
Hi.
Hi.
Hello, good morning.
Okay. Yeah.
So, the ship is on, will be on 350.
Yeah.
Just
Yeah,
bring yourself
we have
in a
the
bit.
time.
And we're going back over to the east then.
Okay.
Just the right.
Okay.
There's a small little forward on there as well.
Okay. Cheers.
I hope there will be a comment on what kind of fish we're looking at, because I don't dare to say anything.
Mm-hmm.
But
Me either.
these dark spots definitely they're not random, sort of, because we've seen a couple of them now and they all have these spots.
Yep.
I think it's the first time we're flying with Will, or at least I.
No, I've been flying with
Aye.
Will before.
Really?
Maybe you're not there.
Maybe I wasn't here then.
It was during maybe one of the first dive, maybe the bamboo coral.
Yeah, maybe
dive.
I came. Maybe I came
Yeah.
later. I don't remember.
We've had now so many dives.
It's been a while. Yeah.
We're on long shifts really, aren't we? Normally you go off at 2, right?
Yes. The balls again.
Oh, there again. Okay, we have
The little fluffy balls that we've sampled previously were also there. They always seem to stack up and accumulate at the base of large stones.
but not all of them either. Like, it could be all of the rocks
Mm.
that have these sponge fluffs in front of them.
It's true.
Ah, but that one has it again.
Yeah. It's maybe something that grows as with the current, just like a snowball.
I'm not so sure. I mean,
What is your hypothesis?
yeah.
Slurps are there.
I'm just.
Slurps.
This is of course a valid hypothesis, but
Yeah.
the current isn't that strong here. And then I'm like, and then the size of them is also quite similar.
Yeah. It's very standardized.
So it doesn't appear to me that this is such a random event.
I mean,
driven.
if you have any hypothesis, please put them in the comments.
Yeah.
We'll be happy to read them and get some inspiration on what
Richard,
these
can we
fluffy
continue the
balls
move?
are.
Thank you.
I noticed that we still have a lot of shrimps in the water column, but not as many as we had.
Oh no, yeah. No. On the cliff there was definitely more shrimp action than currently. Oh, there's an amphipod. Look.
Yeah. I think they look very cute when they swim.
Or you will.
I'm good, thank you, Paddy. So,
That's right. That's right.
unused rest. This football team lost
Oh dear.
to a second division league team.
Oi.
Division below.
Lots of gastropods again. Morning.
Shiro, is that that? Shiro, is that that? What? Routine.
Nah.
Morning, Rob. Jody Way. Nice too. Very nice. How are you? Ah, good.
Yeah.
It's on 3 on wrist guards.
Huh?
It's on caught by with 3 on wrist guard.
No, that's slow. It's intermittent. We've got it there.
Is that near the in or is it after you've been on?
Sorry?
Is it
Yeah.
after you've been on?
Yeah.
It's time to lower the.
So there's a watch change here now in the rough shack because the pilots are
There is.
having a shift change at 12. And I think it's amazing that this just happens in the background and the ROF just continues to fly because they are so routined in doing this that you wouldn't notice if we wouldn't tell you. So it's really
That's one from
really routine and all easy. Hello there. You dressed up.
Yeah.
Yeah.
Shall I leave you my seat a little
If
bit?
Yeah,
And Jenny will update you on those fluffy balls
Yes, I
that
will.
we've collected.
I
I don't know if you had a chance to see them on the
Yes.
screen.
Yes, I
Maybe
did.
you can share your hypothesis with us later on.
No, but I'm influenced. I heard yours. Thank you. Thanks a lot.
Hello.
Hello.
I just realized that we have a lot of white gastropods on the sediment.
For Geographic, can we go
I'm
to
actually quite interested in
0.3,
sitting
please?
down and have a peek, close up, and then maybe actually sampling some because I really don't know what we are looking at.
Yeah, shall we? I like the spot that you chose for the close-up. Just now, that was very nice.
Thank you.
Let's move a little bit further up.
Definitely. It's a bit chilly, huh?
Oh, okay.
Now you're cozy.
Good.
Yeah. That's good. So we sampled. What we did was we sampled with a mug and a small piece of sponge that was actually very firm in texture. It was very surprising.
Yeah, I saw this
Yeah.
operation of the arm and
Yeah, that
and
was
the
cool,
sucking
huh?
that was
Yeah,
quite
I think
impressive.
so.
So
I think so
the
too.
expert I mentioned earlier was watching this
Oh,
sampling, and he thinks it might have been an Asconema, but he doesn't know the species.
uh-huh.
That is something I would check.
Yeah, we should. Yeah.
But it's nice to know some experts are watching us.
Yeah, that's really nice. Thank you.
He's living in Brazil, so for him it's he can watch us during the whole dive.
Awesome. I'm swimming underwater.
What's it?
Jackpot.
Yeah.
What's the direction?
Definitely.
4330.
It's hanging out there. Oh, I need another one.
You shouldn't put this in.
Yeah, I know.
So,
But dinner is such a long time ago.
I restricted myself. I was looking forward to our joint snack later.
Me too.
So, what do we say?
Very
It's a phobia.
Yeah,
It's unusual.
it's really crowded.
Jenny?
Yes. The idea is to keep going to the northwest.
Right, yeah. Well, I'm going off what you said.
No.
Right, okay, thanks.
That's going to drift, you know that much.
Right, okay, alright, leave it to it.
Yeah, get that.
Usually good.
There's nothing irregular going on, it's just a ship change.
Everybody gets on here. Just keep on it. I think it's temperature.
I think it's near massive.
So still, we see,
Can we sit in front of that?
we can sit anywhere.
Can we sit? Can we just sit down so we can look at the rock in front? The rock in front of it.
Do you think it's,
But it's more interesting to look at a rock. And you wanted to sample the snails?
yeah.
Up here, this rock here.
Yes, please. Thank you.
It's not such a show-off rock, as you just said.
No,
You can just
yeah. No, pick your rocks.
Oh, yeah.
Mm.
Okay.
What is this round thing? Is that this colonial Assyrian maybe? Hmm.
Yeah,
Like that.
we're in there since about
With a shrimp coming in from the right.
Do we sit?
No, just stable.
well, rather than
Tell me when you're stable.
Access down.
Okay.
Yes. Catch up.
Sorry.
Yeah, I'm just soaking it in.
So we stopped to once again zoom in and look at the fauna. Look what's here. Take a peek because it looks a bit barren, but it's not. It never is.
Yeah.
So that's a ridge up here.
It really isn't.
So you see the boat sitting on this one?
Yes.
So the big snail
Yes, beautiful.
sitting on it?
It's really nice.
Is there another one poking out? And then you have the Sicilians.
Maybe. We have Assyrians.
And we have the little amphipods crawling to the
Yeah.
left.
Yes.
A little anemone.
Where's the anemone?
There.
Ah.
Yeah. Right to the left of the right laser. The little crawling ones, very hard to see. The red eyes here, these amphipods, they're tiny.
They really are.
Yeah. But well, the one on the lower part, you can see, it's just crawling out of the screen, or just about to crawl out of the screen, right in the middle. Okay.
But how about these little snails?
You want a sample?
I'm a bit interested in them. I have to say, I don't really know what they are.
So,
Can we have a look at them? Just
You're going to get a picture of me down with them. I can see in images. Different stuff.
it's very hard to see because they're very bright. And there are many.
Strand cord.
Maybe we slurp.
the applications were
Yeah, if
Slope
not,
them.
Yeah.
really good.
I think I would like to take a sample with a slurp gun of these little gastropods.
How many slopes have you got left?
I'll just have to figure out where to put it.
Can we do numbers?
Yeah.
It's going to be left.
Those ones would be great. Exactly, the two white.
Or I mean,
Are we too far away? Are we too far away with the arm?
No,
Okay.
I'm just thinking because it will get a bit sedimented.
And that's what I wasn't sure.
Two, isn't it?
Hmm?
Is for tank number 2?
Just a second.
We're just debating because we're not quite sure if we can put it on top of the sponge that we already sampled. If this will influence it. Are we too far away?
You can put it in a.
The same.
Yeah.
Because it's so different from,
Okay.
and it's not far away from the same sediment,
Yeah.
so we're not mixing.
It should be fine. I was just thinking about that the sponge piece might get a bit dirty.
But we already scooped this up with some other.
No, this was the one that we picked. It's very clean.
Oh, okay, then not in
It's
that
very
one.
clean.
Okay, then slope 3, please. 3, please.
Stop. What is this? This is a polynoid. And a sublette.
Yes.
Ooh. This bivalve is interesting too. They are tiny, tiny, tiny. Okay.
You see the tiny sea star on the back?
Yeah.
Very small.
Yeah. Yes.
Thank you.
I put my number in.
Thanks.
It's only street here. Both of them?
Yes, please. Are you fine like this? Or?
Yes, the two white dots. Let's try and not suck in too much sediment. Let's see if we manage.
Just see how you go. And then
Okay, pump them on.
yeah, you've got one.
Got one.
Yeah.
Two, pump up.
Awesome.
Maybe we can just give in the water column when continuing a little bit of the sucking so that they
Goes all the way
definitely
through.
go through all the way from the hole.
Yeah.
Yeah.
Keep on. Okay.
No, that's a
Is
good
it
thing
in?
to just
Yeah.
put it up a little bit and keep on sucking.
It's number
Very
3,
good.
right?
Number 3. Are you
That
taking
sucks.
notes of?
Yeah.
Okay, do you want to, Jenny, while we're sitting here, do you want to take another of this because it was so tiny? Do you want to go on that one?
Yeah, there's two in that.
That's a bivalve.
Yeah, I see that.
We can take that one too.
Or only if you really want it.
I don't know what it is. It can be anything. Could be a new. I
This is so. Okay,
It looks
while
very
we're here, can we put this bivalve, this little shell, into the same number 3, please?
It's on the right of the rock. Yeah.
It's just left of where we.
Oh no, that one there. Oh, yes, that one there. Well, no, that one there. That one
Yeah.
there, yes.
So the reason why we also decided to sample this bivalve for shell, mussel, whatever you want to call it, is because the shape of its shell is very interesting. And I would really love to identify this under the binoculars to be able to say what under the stereo microscope. Sorry. to say what kind of
You know, if you finish
species that is,
get it, you get dim.
what it belongs to.
So you're not running with a ratchet. A little bit faster, easier.
Yeah, there we go. Thank you so
Come up
much.
into the water corner a bit, yes. Let's go in.
Okay.
Yes. This is the
Now that we were here, excuse me, can we just zoom in once more? Jenny, let's find some other words while we're sitting so nicely.
I'll get another section. Section on again.
Yeah, that is very nice.
Zoomed out. I can't go any further.
Okay.
Go ahead, Antonio, eh? Go ahead.
Right, I'll see you later. I'm going, right?
Okay, see you later.
See you later.
Thanks a lot.
See you. Oh,
I'm going a bit closer so I can
Okay.
I've been saying for a minute.
so I can potentially spot them better. The zoom is further along. Can you see this one on the right here? The tiny red one?
oh yes, I do.
It's really tiny. Oh, that will be a challenge.
New merch on already, well.
Sorry?
The new merch
So-
on already.
Are you up for a challenge?
Oh, yes.
Yeah.
Sorry.
Are you up for a challenge?
Right.
It's small.
That's right.
Yep, it's red
This-
and small. Yep, it's between the rock and the stick-shaped
Okay,
wood.
punch it together.
Yeah, I'll show you. Yep.
We can put it together.
Yes, definitely. Put in the same slurp. It's
I can, if it's easy, I can pick up and move to the right.
it's
Just have it.
yeah.
It's so tiny I can almost not see it.
Yeah.
Incredible what we can do here.
Yes, very good. Very good. Mhm.
Start, okay, section on.
I cannot see it.
So small, that's hard to say.
Got it?
I think you have to.
Yeah,
Okay, come up. And then keep the section on. So we
I think it worked.
Section so
Thank you very much.
No.
I'll keep it on.
No worries, but we'll find it in our sieves, and if it's new, we'll describe it. Oh, and then it will have a name.
Oh no.
No,
Oh no. Oh no.
Wait.
That
Okay,
one
thanks
can
a lot.
stay.
Thanks a lot. Do you want to look for one more, or shall we
it's
continue to
all
fly?
good. All good. We can continue.
Thank you very much.
Okay.
Yes. Okay, let's continue.
Okay.
We can sample other things towards it.
Hmm?
We can sample other things in the slurp 3.
Yeah.
This amphibole will be so tiny that will be hard to find.
There's a tumbling ball again. Paco is very interested
This is
in
the
this.
trajectory they were on.
So I will just zoom in on this for a second.
Yes, I can see it. It's in the middle, and now it's going to the left corner of our
Corner of
screen.
the dog leg.
Yeah.
Try and get the outlet and then go on the bar next west.
Because they are piling up.
And next one.
Maybe he's right. His hypothesis is correct because he said that it's possibly sponge spicules that roll until they hit a rock, and then they just lie there, and maybe he's right about it.
It will be easy to find out once it's on deck.
Yeah.
Right.
It's a
very snackish day.
Yeah, it is. Sorry, I just wanted to center it.
Oh. Sus.
I wanted to write something down and now I forgot.
Sista.
Okay,
Yes.
Hymenasta.
Was a little glass sponge.
Yeah.
Start moving north. Maybe.
It's a side
Are those burrows or rocks?
Bridge ROV.
Soft core.
It's hard to tell.
Yeah.
There's a dark spot. We don't know if it's a burrow or like a hole or a rock.
I think it's rock. No,
Yeah.
it's a rock. Okay. We have a tape change in 10 minutes, alright?
Okay.
Sure.
Bridge ROV.
Hartley Bridge. Sorry, Antonio, I had alarms here and needed to leave it.
Okay, no problem, Paul. So we are ready for the next movement. The intention is going north at 0.2 continuous.
Or 0.2 continuous, okay?
Thank you.
Sometimes there isn't really so much to tell, huh? Apart from a
Nice
very
to look.
beautiful soft car on a rock. And another whelk, which I very much appreciate.
It's a soft sediment. Some rocks distributed.
Mm.
Really different fauna to one observed before. 1,200 meters.
Yeah, it's completely different.
I had expected a bit more. Yes, from soft corals and anemones, but I assume they'll start in shallower depths.
It's a nice sea star. Sorry, is the aim to
I'm
move
going
to
across, yeah.
northwest again? Yeah,
Yes.
yeah.
Yellow line.
Okay, thank you. This is a different species from Hymenasta. That was something else. I am completely missing ophiurids and sea pinks or sea cucumbers. I think it's too shallow, maybe.
Yeah, and the other theory was that there are too many sponge spicules from these mats, and then it will be quite tough on the cucumber. Maybe they don't like it because
Yeah,
they
yeah.
digest all the sediment.
Yeah, you're right. It's not their type of habitat.
I've just brought around from
Well, because there's so many sponges. Dust sponges.
Yeah. True. There's rocking. It's hiding. There's one again.
Yeah, and shrunk.
I wonder what species this is.
Yeah. You see
I know
the
it's not
directly.
enough that way.
No, asterias of some. Hard to tell. Lots of snails. All the white little dots are snails.
Oh, that little white things.
Yeah.
They're all snails. And we have a nice little group of glass sponges there. Rostily there. Aw, look. A baby humanasta on the rock. Aw. That was cute. And some soft corals. Probably gazemia. Not so sure. Maybe. Ooh, quite a large ascidian. Okay, I see some boulders. Maybe we get a bit of more hard bottom now. It's like what we said in the beginning, that you can be very precise and detailed with the transect planning and look at the profile and try to estimate whether you will have a soft bottom or hard bottom and about the slopes and everything, but in summary you basically just have to dive down and see what you're facing. And this seamount is currently catalogued as an unnamed seamount in our database because it has no name yet, and we believe that no one has been here yet to explore its diversity like we do right now. And what is in particularly interesting about it is that this seamount is on the eastern side of the mid-ocean ridge, of the Knipovich Ridge. Knipovich Ridge, my god, that's hard to say. And along this ridge, along the Mons Ridge and the Knipovich Ridge, we usually have lots of seamounts on the western side, but not so many on the eastern side. So in order to conduct comparative studies, this is a very interesting site because we have different types of water masses that flow in the deep sea on the eastern and western sides of this ridge. So we would expect, or there could be faunal changes and different types of communities that live at the same depth but are influenced by different types of water masses. And what we for sure can see, that this is a completely different ecosystem than Schultz Bank, for example.
Yes, for
Like
sure.
this is
That's why it's
not to be compared. Like, in that sense.
That's why it also takes a few samples to get the sequences, to compare the molecular.
Exactly.
And then we can look at sequences of also of the same species to see how much they differ.
Oh. We have a tape change in 1.5 minutes.
don't think this is moving.
Yeah.
I think it's holding.
You think? Aye. What's
this thing?
that?
What's this hanging in here, right in front
Is
of
it
us?
a fish? It's a squid.
No, it's
Can
a
you
squid.
slow down, please?
Mhm.
Let's slow down, please. We have a squid and a tape change. Okay, everything at once.
Oh, thank you.
Oh,
Well,
that's
that's
okay,
the
yeah,
sixth.
And we are pulling out the sextant too. Well, how do we do that?
There are some slight technical things. We only have one rough pilot at the moment, so please,
Oh, we're
please
okay, don't worry.
Yeah, do you know how to pull out the sextant? And maybe can we come to a stop, please?
Antonio, can you do the saxon out, please? Just hold that position.
Thank you.
Do you wait to land or just?
No, just
No,
a stop. No,
it's-
just a stop.
This is beautiful. This is really beautiful.
Okay.
We need a
Um,
tape change.
We need a tape change, and I would love to dim the lights, please.
Oh God, everything at once.
We just check. I just want to come a bit. Yeah, it's fully out. So like this.
Jetzt
Ja, das fängt gleich raus. Der geht das gleich raus. Besser?
Yeah. I will look.
Can we turn the rod a little to this? Can we try? Mhm.
There it is.
We will try and get a closer look at it to
Yeah.
better be
Yeah,
able
sure.
to identify the family maybe or the genus of it. This is a beautiful footage.
Yeah,
Three.
that's
But it's difficult. Ah, it's still there. It's with us.
yeah, but it's going under the camera.
Ah, right, okay.
There it is again. Just follow it. It's fine. We're stirring up a bit of sediment here now.
There
Sorry.
you go,
we-
thrusters to keep us off the sea.
Yeah, that's all right. That's all
Yes,
right.
absolutely. Sorry.
It's
Can-
very difficult to follow a squid.
And sorry, can you do the tape change, please?
You want to do it now or wait for this?
Yeah, let's wait.
images?
Let's
I'm
wait,
very
let's
happy
wait.
to wait for after this.
We have a bit of storage on these tapes, don't we? More than 2 hours.
Yeah.
Wow. Thank you for this. Beautiful show.
I'm at the end here.
Ah.
Sorry, zooming out again just because we're losing it.
It's a bit difficult to maneuver with the camera and really. Yeah, but this was lovely.
Thanks a lot.
Maybe it will come back down. Maybe not. Okay.
You want to do
No,
the
no.
tape change now?
No,
No.
it's
No,
back.
sorry.
Okay.
Maybe it's swimming towards us.
Let's
I
just.
don't know what it's doing. Maybe not.
What is it doing? It's definitely interested in us and the lights, and now it disappeared, I think.
Yeah, sorry.
But that was
Okay.
beautiful.
We'll do a tape change now.
We'll do a tape change now.
You don't want it to come back, be in full view and not have it.
Do you want me to continue along?
No, let's just wait for a second. Please.
And recording again.
Okay.
Okay,
So
we can
now-
continue. Thank you.
We have seen a squid, changed the tape, adjusted. You know, there was a lot going on now. So let's see. Yeah. This I will log that this squid was of the family Omastrefidae.
Yeah, that's just about too much. Just,
Sorry,
yeah, I thought you just entered Squid.
I did. And now I will update it to Omastrefidae. And we actually have a squid expert in Hamburg. He's our colleague. He also works for Senckenberg. His name is Alexei Golikov, and I'm sure that he can help us to identify the squid
Yeah, me too.
to genus or even species level. Oh, nice fish. So we will figure this out. What kind of species just visited us, or which species we visited with our RV? Because we are basically.
Look, it's going underneath this. Look, this fish is going
Oh,
under the
yeah,
sponge.
he's really interested. I believe he wants to eat some tiny little amphipods.
Sorry.
No worries. Oh, Asconema is coming. Not Asconema. Asbestopluma is coming back. Does it get a bit more rocky?
Could be.
I think so.
Goodness. I would like to, not for this dive, but I'm more interested to see the other side of the mountain. This is an elongated shape. It's an elongated shape. So on the one side, and I think to go over the side and look on the other side, it's maybe more into the current.
Yeah. Do you think the signal is back or no? Okay. That was a really nice moment there with the squid. I liked it. It was, it lasted for some time. It's lovely. Yeah. Can we pull the
When we
sexton back in?
But that's for them.
Maybe?
Do you want to go up this ridge?
Yes, please.
Right. Side count.
is this? Can we stop? Sorry. I saw something on the bottom, right? Like,
Yeah, yeah.
Okay, we're on there. So what? You're gonna have to point out
No.
to me,
No, but
No, no.
we were too late. Sorry.
Never
We'll
mind.
Just continue.
never mind. All good.
Sorry, I'm looking at different screens
yes,
here.
we
I'm
know-
looking
we know.
down.
No worries. All good. Now we're back.
And we're
Putting it
on
back.
the ridge.
Okay, we already have the updated positions. so
Okay.
Perfect.
We are there.
Oh,
Jolly.
10 of 4.
Perfect.
Very good.
So we're right on the ridge.
Yeah, this is more. Now we have a change again in sediments,
Yes.
and we are more rocky. We're
What-
back to
what
the.
Yes, yes.
I would like to go
Agree, Jarvi?
a little bit on the right side of the ridge.
On the right side?
Naja, ein bisschen danach rechts.
Wir kommen doch gerade von rechts.
Okay, sorry,
I'm gonna
Paul.
check.
We would like to change the direction of this movement to 035.
Können wir vielleicht auf der Ridge weitermachen.
Also wenn überhaupt würde ich hier tauchen.
035 at
Nicht
0.2.
da, oder?
Okay, that's it.
Oh, it's a feather star. I thought it's a kind of arm again, but it's a feather star. Very nice. Have you looked at soft coral?
Yeah.
And the fish inside the sponge, did you see it?
No, I didn't.
It's sitting inside this big glass sponge.
Sea star.
Lots of crinoids coming up.
Yeah. And another rockling. They really like this habitat. Looks very interesting.
Yeah, it does. It have a slightly different shape than the sponges have many shapes, but this one had a lot of thin parts in between.
I But yes, we have a change here now from rather continuous soft bottom to hard bottom again,
Can you get me a bearing? That's all right.
and we are flying along the ridge,
Two red and green.
along the crest of the seamount. That's beautiful.
Side points.
I don't like that it's so far.
No, me neither. But then move the camera back in. That's what I'm trying to avoid.
Yeah, but we can ask them.
Yeah, can we fly a little bit lower to the seafloor?
Yeah, I'm
Is
coming
that
down now.
possible?
once.
Thank you.
And maybe also retrieve the sextant because
But that's up for them to decide on the trident.
thank you. All right. Do you think there are any sharks here in this area? I have no idea. Like whether it would actually like if it would actually if it would be possible to encounter like a small shark. I've seen one when I was diving once. That was really cool.
I wouldn't mind.
Yeah.
Greenland shark, for example, but
Yeah, I was
it's
thinking more
extremely
about
unlikely.
I was thinking more about like small shark species, benthic, that like to just lie on the sediment. Maybe not. I don't know. Just the water is a bit murky.
It is. We do have a rock coming up. Maybe we can stop in front of it.
Which of them?
Well, I thought it's not a ridge, but it's not. It's so flat. You saw what I...
When you said rock.
yeah, I thought this would be a rock line. But it's not. It's
no.
just a different shade of sediment.
So I've been logging soft corals again, although we don't have that many. We see them. And we still see feather stars on these glass sponges. And we're back to a bit more of a soft sediment bottom. So it's changing quite a lot on the seamount.
Some acidians.
Could I see the... Is it possible to remove that screen from the mapper? Thank you. Can we zoom out a bit from that view? Sorry, I didn't mean you. I actually meant the
pilots for the map, yeah.
Yeah. Okay. Let's check in. And decapods, maybe.
They're coming in.
They keep coming.
No idea. It's actually burrows or
Maybe.
stones. It's really hard to see
Yeah.
in the light. And
Yeah. No, it's stones.
Because these
Stones.
Yeah, these stones in the bright light, they have a very dark shadow, and from the distance it's impossible to see if it's
Yeah,
a hole in the ground or
yeah.
reflection of it.
Ja, med nästa.
Lollipop?
Is it?
I don't know, I'm guessing.
Shipped to speed.
I'm guessing whelk, actually. I'm
Rich,
guessing
are we-
whelk.
Can we increase the speed to 0.3?
I can see the siphon. I think it's a whelk. Maybe not. No,
But the siphon
no.
being as long as it
No.
was, I think it's unlikely.
no, it wasn't.
I think it was a lollipop that was bent over.
And then
Okay.
this USB was on the back, I took it out, bring it here again. It's still frozen and only came back again when I changed on the GPS sector to COM4. It was on COM10.
Yeah, I think origins and contents
They were working with COM10 because
Sea star.
they were having positions, but
yeah.
Feeding.
now if I change to COM10, it stops receiving. Only on COM4, which
Oh.
is the one that we used last time, at least last two.
They are nice.
I think how do you say concussion?
And they do, all of them are spotted, but they all have a different pattern. They're really nice.
Now it's working again with a different comfort.
Look at this vast area. Wow. Sea star again? Yeah. It is. So this is a truly exploratory dive along a transect that we planned on the ridge crest of this unnamed seamount, and as you can see, we are exploring a highly sedimented area. And currently we're seeing sea stars of an unidentified species. I cannot even say the family or the genus. I don't really know what it is.
But our experts will know.
Definitely. These data will be analyzed and annotated at a later stage. And even the sponges, like the density of sponges really declined now too. So we observed a change here. But then again, they appear again in the background, so. But we have a lot of snails on the seabed. Sometimes also the buccinid snails or whelk-like snails. I don't know what that is of a sponge. There are small anemones.
It's not really helpful.
It's a bit quick if we have it that much zoom, but
Yes, please.
maybe I will change slightly the direction.
Yeah, just
Towards
The anemones are quite nice.
Bridge Alvi.
Okay, great.
Okay, Paul, can we increase the speed to 0.4 and
Spanish.
change
Oh.
the direction to 0.60?
Wonderful.
Mm-hmm.
060 at point 4.
Okay, let's see, thanks.
The work's really hiding. It's cute. And a fish.
Yeah, I think so. I have no idea we're doing this.
Oh, Humanasta.
Probably to do because it's
So-
here, so to
I'm pretty certain this is a Humanasta sea star, but we have several species that live in this environment. They're not all Humanastas. We have a
And they're very slimy. So when you
I
put-
big look on it, and that was too late, I think.
Too late. Yeah. Just log it as
Yeah,
too late as well.
yeah.
just roll
Looking for the Sumanasta, they produce a lot of slime. And it's slightly irritating when you have a lot of samples where you want to take DNA from. You have a little slime cover on everything.
Yeah, I agree.
And that separation was already there. And you can see the lines.
But already zoomed in quite a bit, so if you see something, we have to stay or stop immediately.
stop the other beam, keep the vessel moving a little bit more, because this configuration a bit.
What's that? I think it's carnivore sponge on top of the stone. Looks like asbestopluma. No, maybe not. Huh?
Too late. I'd like to see that.
Yeah, we're a bit late.
We're really a bit late. We have to zoom out and trust ourselves to.
Yeah,
focus.
don't zoom that much
Yeah, no.
out.
I was a bit overdoing it.
Stopping this.
We sometimes see things, but then
No, we're too late.
then it's already too late. Then they're all
Well,
of
it's
them.
there.
Flying just keep going.
We can stop in front of those guys, or
Yeah, it's true.
can we stop to the port side here in front of those sponges? Would that be possible?
That sponge, you can see the top left corner. Yep.
This one's coming up here.
Seems to be perfect.
Oh yeah, it's a crinoid. So we're having another stop at the seafloor to just admire the feather stars on this very interesting growing sponge. A glass sponge.
Like little arcs.
Yeah, it's really, really quite cool.
This one I like that, yeah.
Okay.
Do you want me to sit down next to it?
Yeah, just
Yeah, or stop. Whatever's best for you.
yeah.
Things I'm close to the sea edges, so that's what I'm thinking. Oh, this. Do you want me to land?
Yes, please. It's easier. easier to settle them.
Yeah. Look at that one too.
It's too far.
Yeah.
Is this one or?
Yeah. Yes. You're good. You're good where we are. Landed.
Yes, I'm not.
Okay.
Thank you.
I'll first zoom in on the back on this little red one.
Aww. It's so nice. I tell you, I don't really know.
All these worms.
Yeah.
You see it?
Yeah.
All on this one
Yeah.
side of the. Do you mind pulling in this section?
Sexton is going in.
Thanks a lot.
I really don't know what kind of sea stars these are. Wait. Oh, what are we having? Oh, I like this. Yeah. It's a gastropod. Isn't it?
Yeah, can you turn off the lasers, please?
Bridge ROV, can we stop for now? Stop.
We have a glass sponge with a gastropod. And several feather stars. It's really beautiful.
What is this little pink thing? Is that an anemone or is it the base? Or
That's another gastropod.
it is another gastropod, you're right.
Can we zoom in more?
Yes, just a second, what is this? On the right
Bryozoan.
side?
I think it's a
Getting
bryozoan. Yes.
I don't want to be super annoying, but what's this little red thing crawling?
That must be an amphipod.
Well, I give you 10 points.
Thank you. We haven't had this before. Gastropods,
The
on
gastropods on the
sponges.
sponge. No, we
That's
have not.
new. And it's nice to observe this. That they actually sit on the glass sponges. So these feather stars, they're also called crinoids, are sister to sea stars and sea cucumbers, sea urchins, and brittle stars. And if they wanted to, they could just lift off.
Look to the left.
Yeah, I don't know. What is it?
I don't know.
Zoom in. Let's have a peek. And the sabelid in the background.
Sabelid in the back, but there's also this.
What are we looking at?
I don't know.
Me neither. I think it might be an ascidian on top of something sponge-like.
It just doesn't look like an ascidian to me. I don't know.
It looks very funny. There's a baby
Is this a
humanasta.
larva? Is this a larvae? You're talking about the Caesar. Oops, sorry. I didn't
Yeah, I'm
mean to-
sorry.
Yeah, just-
Look.
Yeah,
On
I
the
saw that.
sabelid tubes.
I am.
Mwah.
The sublip tube here, that's a worm tube.
Yeah,
Here there as well.
it's tubes of bristleworms, and they have the same
Oh, it's, sorry, it was so zoomed in. It gets a bit clunky to move.
There's some current here. Look at it. Wiggle.
Shivering.
Yeah.
You see the sea stars?
Yes.
Yes, but there are quite a few. There's 1, 2, 3, 4.
The baby
Yeah.
amphipods? Did you mean the amphipods?
No, I meant the sea stars. Yeah, well, the amphipod is right in the middle.
I just see.
The amphipod is the one crawling
I
towards.
see 3 amphipods here. 1, 2, 3,
Good.
4.
Okay.
Where are the sea stars? Sorry.
Am I mistaken?
Yes.
So
Sorry.
little red.
That's. I don't know what that
I
is,
can't
but.
But it has a similar
Mm.
Okay.
But this is interesting.
Yeah. Let me zoom out first because it's so hard on the eye
Mm.
to move. So we're moving to the right of our little sponge. And we'll zoom in here again. Going lower down, just to be able to get as close as we can.
So there are bryozoans in the sediment. I've logged
Yes.
this.
Yes.
It's quite many.
These tree-shaped units that are brown and white. What are they? What's an English name? Lace animals?
I don't know. I just know them by bryozoans.
A colonial organism, many little units. Hi, Marco. So we can continue. This one?
Yeah.
Whose is this?
Frozen Jenny? Me, I brought all kinds of clothes and blankets here today. We can continue.
Continue, yeah.
Oh, unless Paco wants to.
No. Please
Okay.
do.
You want to sit here or do you want to take the camera?
I'm okay here.
Either way.
Do you need anything?
No, I'm good.
Okay.
Okay. Okay. Line up. It's quite sandy. I
Did
don't
you
know if have
did
watched.
you collect anything
Reach
more?
Ivy.
No.
Well,
Bridge.
we just we
03.
collected, but
Yeah.
I'm not sure if it
Okay,
was.
the next movement
We had
is direction 040
Ah,
at
okay.
0.3
No, I
knots.
wasn't there. No. Cool.
Okay.
040 and 0.2.
That's it. Thanks.
Ah, there.
We should leave 2 slopes for the other shift. Otherwise they'll be
Yep.
not impressed with us. So we can slip something on top here.
Yeah.
And we didn't dare to put this on the sponge. But we should keep them.
Yeah. Yeah, there's already here. There's already quite a lot. Ah, okay. Perfect.
I'll have a tea, please. Do you like? Can you take that? Yeah, just put it up. Give it afterwards. Only it. Just use it. Yeah. Just, yeah. So thank you. Cheers.
Australia, what name?
Oh, there's this ray.
Right in the middle, yeah. Sorry, I should have said that. Just
Have
looking.
you?
Yeah, no, sorry, I did see it.
But did you see
And,
some before? Because this is the first one I
this
see.
is the first, you're absolutely right.
Yeah.
This is,
And we are at 1,252 meters. Mm-hmm.
yes. Can we turn the lasers back on, please?
Did you take another mug? Or not yet?
No.
And
I thought it would be more interesting to have more distance between them.
yeah.
To have it in a shallower area.
So
be
maybe
able to compare.
for the other group
Yes,
to take.
exactly.
Yeah.
Oh, there's a ray.
Another one.
Oh.
It's swimming into this.
Yeah, probably the same one and earlier. So this looks like a female. The male has two claspers or penises that should be seen near the tail, but this one doesn't have that, those two claspers. So this is probably a female. So on our previous dives, we have seen a lot of Arctic skate rays, especially on the top of some seamounts. The Louis Boyd seamount, for instance,
Yes.
and we saw a lot of eggs.
A lot of eggs. That's
But on this side of the Mid-Atlantic Ridge, this is the first
the
ray
eggs
we
of these
find.
rays are quite characteristic.
Mhm.
So normally, fish you can't see the eggs in ROV.
Yeah.
But those are rays and of some sharks. They've got this characteristic shape, rectangular, with extended edges.
Yeah. And they're dark.
Yes, and they're easy to spot.
So currently the diversity seems a bit less than on the cliff.
Of the megafauna, yeah.
Yeah. When we at the very beginning of the dive, there was we landed on a cliff that was extremely rich and dense in sponges, soft corals, anemones, tube worms, etc. And then we arrived on this plain with boulders on which there are still sponges, but of course it seems less dense
And less colorful.
and less colorful. Oh, there's one of your amphipods.
They do come in sometimes.
Hmm.
There's another one.
We're on an exploration dive. No one has come here before us, so we do not know what to expect. We've tried to decide on the transect, on the path that looked promising, on a map that we had, because this seamount has been mapped, and we've tried to use the map to decide on what parts to explore, but it's always kind of a gamble. It's difficult to know what's going to be soft sediment, what's going to be hard substrate, and now with the knowledge that we're getting from this footage, this will be useful for the scientists coming next to this unnamed seamount. They will maybe want to re-explore the cliff that we discovered, or maybe return to this plain also to subsample the mud.
You want to stop another time?
Yeah, I will tell you. If we see a
This is better.
pompon sponge to sample for population genetics, that could be interesting
Way to that
to stop.
point.
But I enjoy the same photo. So
So it seems we have a lot of viewers in the US right now, I've heard. And we didn't have that many viewers in the US in the previous dives.
Well, it's now 1 o'clock in the morning
Yes,
for us. So.
but it's nice to be able to speak to a completely different audience than our previous dives. And so for our viewers, we are in the Arctic, in the Norwegian Sea, and on the eastern part of the Arctic mid-Atlantic ridge, on an unnamed seamount.
1,246 meters.
Yeah. So the remote operated vehicle, or ROV, is below us. We're on the ship in the middle of the ocean. We're currently in the command center, or ROV shack, which is on the deck of the boat. It's a bit chilly, I have to admit.
I put on an Antarctic suit, so
Yes,
I'm good at
actually,
now.
that was a really good choice.
It was so
I
cool.
regret I didn't have that idea before replacing Jenny. But I mean, the reason why it's cold here is because we need to actually keep this room cold because all the electronics.
And we're sitting under an aircon.
And we're sitting right under the AC. Yeah. And we've been streaming live now for several hours, so of course we're starting to get tired. We're waiting for our shift.
No, it's all good. We just repeat ourselves and getting a bit uncreative.
But I think maybe the two scientists that will take our shift might arrive at the level of 1,000 meters. Things might start to change.
But they will.
They will
Yeah.
maybe see.
It was the other way around
Mm,
the days before, but it will. We assume it will get more colorful and
yeah.
more different, larger animals when it goes to 600, 700 meters.
Mm. So according to the map, we're gonna get to a point where we take a left turn at some point, and we'll be on a kind of ridge, which looks promising from here. But again, our maps are not very precise. This mountain has been mapped with a resolution of 35 meters, which does sound promising and good, but still it's not the resolution of 1 meter that we have for other mountains, other seamounts. So we always have this level of uncertainty that prevents us from knowing exactly which path we should take. So it's a lot of white sponges that you see. These are glass sponges of the family Rossellidae.
And often you have feather stars sitting on top.
Exactly.
Also filter feeders, trying to get a little bit higher into the current. Got a sea star here. Right in the middle. Just
Oh yeah. Okay. Yes. What is? Oh yes. We have also a few ascidians, these transparent bags that you see attached to the bottom. These are also filter feeders.
fish.
Yeah, actually that's one of the things that is more abundant here is fish, I think, compared to our dive yesterday. Oh, this looks like
we would like to,
huge.
to look to this big rock on the left, please.
Yep.
But we're getting to that point where
We're starting to climb.
Mm.
Wait a bit just to see.
So please stay tuned, because I think this is going to get a little bit more interesting, maybe with
And we hope
a few more
vertical meters.
larger animals. I'm gonna log this. Let's see.
So we can't get any closer here.
We'll increase the range to 20 instead of 10.
Yep, yep, that's right.
Look at that.
Are we flying up here? Are we flying up or are we stopping? What are we doing?
The thing is, we're coming over some of it.
Oh, okay.
So I'm trying to keep my crashing to the seabed.
So we have a kind of cliff. With a lot of worms again.
Soft corals.
Soft corals. A lot of soft corals. Wow.
What's this?
Anemones maybe? sure either.
Mostly stuff.
Oh, okay. So it's not a very huge cliff. This
no,
is
it's not a cliff, it's more boulder.
yeah.
Bridge RV, can we reduce to 0.1?
Oh, wow.
6.1.
So I can go down more if you want.
A little bit lower.
So it looks like a little mound, or I mean, really. Oh, there's fish as well.
Go down more.
No, because it's getting so muggy. Can we just stop here? Maybe it will settle. We'll just
Yeah,
give it
well,
a
we need
couple of
water
seconds.
here.
At the base, you mean, or?
Yes, we'll go down lower.
Okay.
But this is an overhang, so we can't go in there. Otherwise, we'll go underneath it.
Okay.
Okay? So, keep this here, down a little more.
Okay. It's quite a nice little over.
Yeah, it's very nice. So water temperature, let me see. I like to check the water temperature. Minus 0.2.
Okay. Do you think this will settle? Sediment? Or is that
Because I'm
current?
using too much thrust.
Okay.
Water. That's the screen I look at, not yours.
Yeah.
I don't have the luxury panorama like you guys. So I'm trying not to go into that rock.
Of
and
course,
I can't
of
go.
course, of course.
Of course.
You're doing superb, so
Yeah.
don't worry. Well,
But I can see there's some geodias, there's some carnivorous sponges, so here we have a bit more diversity.
yes. This is also a slight overhang.
Yeah,
So
probably a lot of
challenging.
currents.
Yeah,
Yes,
challenging.
challenging for the
Oh, yeah.
pilot and the ROV. But those large, hairy, brown globes are the genus Geodia. These are sponges. The feather-like or Christmas tree-looking animal is also a carnivorous sponge.
Okay, let's
Oh.
let's continue the yeah. Shall we continue?
Yes,
the flight?
we can continue because
Thanks a lot.
it seems a bit too complicated.
No, and maybe we encounter another.
Well, I can
Yeah.
back
Another-
off and then it won't be
another rock.
affected by
No,
the
because
thrust. And then you can
Okay.
zoom in. The things are going across.
Yeah, if we
Okay.
travel
By the
from
thrust.
okay.
at a distance, that's also good for us.
And then you can zoom in better. And then it won't be affected so much by the thrust.
Bridge RV, can we stop for now?
That's fine.
Yeah, so it's
Let's fly on, and then we can find another nice spot.
You want to pass the RV to the other side? No.
I have difficulty to understand how this
Me
is.
too. Me too. I didn't
yeah.
see it as an overhang until
No.
I saw the other cameras. It's
Yeah.
very hard to get it three-dimensional.
Yeah, and because of the poor visibility, it's a bit complicated to see how far this goes. So we're gonna try to fly above it and then reconnect. There seems to be a series of boulders or little hills. but we're definitely getting out of the flat area.
Sometimes big messages from the dry lab or from people writing and whatever different channels is watching, and some are quite excited about the squid.
Oh yes, I saw that you saw a squid. Yeah.
Yes, but we will share the images with our colleague who's an expert.
Aha.
So I'm saying it's most likely new because it had certain characters.
Uh-huh. Okay.
I know too little about squids to judge that, but he will.
There was another
Nice.
comment, actually, there was another question asking if our lights produce heat.
Oh, that's a question to the
Yeah.
lofty.
But
LEDs, so
exactly,
minimal LED.
exactly, that's what we answered. We said the LED lights actually know very limited heat.
Older style ones like halogens would generate about
Mm. So if you've been wondering, no heat. Oh wow!
Very barren landscape, and as
Yeah.
soon as you
So-
over this boulder,
so there was
looking
like
down
a-
on the other, also on the other camera, there are quite a lot of sponges,
Yeah.
going down. We'll continue going straight, a little bit high up this ridge.
So we're now soon starting the last part of our transect of this unnamed seamount dive, which means we'll be live for another couple of hours, but this is the last part of the transect that we have planned. And we are back in our sandy flat surface.
I had hoped it would changes.
Me too.
But now-
Me too. I thought this was the end of it, but no, it was just a few.
The sediments are interesting when you look at the creatures at the microscope, but looking at it in this distance, we don't see anything smaller than centimeter. So
Yes, of course, you have to imagine actually all the diversity that's inside the sand, but
that is not.
from here, from that distance, unfortunately, it's not as exciting. I should remind you that our lasers are 20 centimeters apart. This gives you an idea of the size of the things that we show you.
I have to correct you.
Oh, it's 10.
It's 10. It's 10
It's 10.
centimeters.
Thank you.
Bridge RV.
Bridge.
Okay, we can keep going with the movement. Direction 040 at 0.3. So let's do
So our ROV pilots have constantly to communicate with the bridge on the boat, because we are connected by a cable to the ROV, and the ROV and the boat need to move together
That's it, only we don't pass
in order
the
to do this
the
exploration.
vehicles will be crossing the
So as soon as the ROV stops, the boat needs to stop, and when we start again, the boat needs to start, and in the same direction as us, as the ROV. Therefore, the communications is critical for this
We're actually
exploration.
sitting in a container on the boat.
Yes, I'm always thinking that I'm with the ROV, but I'm not.
Exactly,
I'm
me
on
too.
the
I'm also
Thank you for specifying. Yes, we're on the boat, but the ROV is below us, and we're
1,240 meters below us.
Yes.
So more than a kilometer.
So I was reading actually that the seamounts that are on the eastern side of the Mid-Atlantic Ridge are a bit more influenced by glacial sediments, because of course during the last ice age, Northern Europe especially was covered with glaciers that went really far away into the ocean and produced a lot of glacial sediments by eroding the bedrock in Europe. And a lot of these glacial sediments fell into the ocean, especially when the glaciers melted. And so there's a lot of really fine sediment in that part of the world, around Svalbard, in the Barents Sea. And due to the currents, a lot of these glacial sediments then end up on the eastern part of the Mid-Atlantic Ridge. I don't know if the sediments that we're looking at now are this type of sediment, but it could be. And it confirms a little bit what I've read, that maybe some of these mountains are a bit more covered with sediment, because I mean the other seamounts we've seen on the western side didn't have as much sediment so far. Maybe we were lucky also to find rocky outcrops, but we'll see. Since this mountain has never been explored, this is just the first glimpse we are getting, and it's difficult to conclude to anything, but yeah, we've seen a lot of sand and mud today. But now even the rocks have disappeared. So before we had a couple of rocks we could stop and watch a little bit the fauna, but now
I feel a bit let down. We've been so spoiled with
Yes.
our dives lately.
It's true.
We had such a diversity on this expedition.
But I mean,
we saw this coral.
the bamboo coral reef,
Yeah.
Schultz
Garden.
Massive yesterday, Schultz Bank.
That was my favorite.
So yes, that is a big difference with this dive. The beginning was quite extraordinary. It was nice, very nice.
We're kind of waiting for something to catch our eye.
But it's part of the exploration. I mean, we have to start exploring part of it, and not knowing which parts will be soft bottom or hard bottoms. Of course, we favor hard bottoms because we know suspension feeders, filter feeders will go to these bottoms.
Come on, it's a sea star and a shrimp.
And a fish here on the left. So
And a feather star.
all hope is not lost. So actually we could
It sounds
collect
a
this.
little bit
it's a bit-
dramatic.
yeah.
It sounds a bit dramatic for.
No. It's just
We're just spoiled.
it's a bit late for us, and but I can I saw a couple of pom-pom sponges, little white bushes, and these I think will have the interest of our scientists coming for the next shift. They will probably want to sample some of those.
Look at this sea star.
Mm-hmm.
Do you mind logging another sea star,
Mhm.
please?
Okay, sponges are back.
And
Cor-
soft coral.
Feather star.
These feather stars, I always climb on top of these sponges. Going into the current.
It's funny because in almost every little oasis, sponge oasis, there's always one fish that's resting, and many of them.
What's that? Was it a soft coral? Oh, that's Is there such a temperature change, or why is it so flimmering? In the
So
water,
water temperature is still -0.2, so it's slightly increasing. But I did see that shimmering.
shimmer as well.
I did see
Maybe
it.
it's a seep. I don't know if you saw it, but the water quality, it suddenly had this,
Yeah.
like when there's a temperature change in
Yeah,
water, when
yeah.
you see that. Could be seeps. I don't know.
1,200. It could be. We could also be between two water masses. Maybe something. I don't
But we're
know.
quite low above the seafloor.
We are. That's true.
We're just getting along. I'm glad you also saw it. It is quite murky.
It is.
It's also unusual.
Let me write this down.
Because there's so much flying in the water, why are there not more filter feeders? Suspension feeders.
Oh, okay. Are we going down or
Still, yeah.
a little bit downhill?
No, we've got a bit of a
Drop off to the
drop
left.
off to the left, so I'm coming left as well. It's closer to the
Okay.
left now.
Okay.
So yeah, we are getting a little bit of drop off.
What is that? Could we slow down and look at what that very large sponge is on the left, just above?
Yeah, let's just stop
Maybe.
first.
Right here?
We don't know.
So, this one?
Yes.
Ledge. Ledge on the left, obviously.
Is that one there?
Yeah,
These two.
So I'm thinking of the really large ones, you see? Yeah. Okay.
I'm not sure if it's easier to sit there off or to move it before.
To land here?
If we can land closer to the really large one.
A large rock on my left, so I'm going to come this side of
No,
Oh, sorry.
it's after. Yeah. I think you're gonna see it.
Ah, right. Okay.
Yeah, there it's coming. There it's coming. Yeah. There's a way. It's a bit complicated terrain here.
You wouldn't think so, but there's
No.
just a big rock in front of the sponge, which makes it a little bit more tricky to land. And of course, we would like to sit down just in front of it, then we can see a lot more when we zoom in.
I expect that it's a beautiful sponge to examine with a lot of feather stars. It could be a nice stop. We're gonna wait a little bit for the dust to settle, and then we're gonna wait for our pilots to tell us when they are also stable and on the ground.
Stop.
You've stopped. Okay.
Then we'll just
Okay.
wait until this.
Let's wait. Yeah. I mean, we haven't stopped for a while, so we don't really know how the sediment is. Is it very fine? And
We have
this
to
is
try out.
we have to try.
We
Yeah.
have to be patient now and try. Patience is not my skill, but
Oh, but look! Look!
yeah, that's getting there.
Oh.
If you take a little blanket here.
Oh, thank you.
You're welcome.
Ah.
Wow, that would be nice. May I play?
Yes. Oh, look at the fish. Oh, this is going to be nice.
Lasers off, please.
Put that up on the side. Oh, you put up on top of that one. It's empty. Thank you.
After a couple of minutes of sand and nothingness.
So, even the stones left us as Paco.
Finally something
Finally
to...
something to look at. You see on the left side here is the soft coral, which we will lose out of the frame when we go in further.
Yes.
And we see these two fish.
Oh, that's an amazing sponge.
But it's not actually the Assyrians.
Yes, I see those.
Oh, don't leave us.
Oh,
Bridge,
Come on, fishy.
ROV, can we stop for now?
Oh, can't shy.
but let's try to see how big this sponge is. Because if this is 10 centimeters, do we have a ruler here? I don't think so.
This sponge is more than a meter.
More
right?
than a meter. More than a meter.
That's is it the Rosalida? somewhere?
No. I would love to have a piece. I think it's a forsythia,
Oh.
but I'm not certain of the species. I'm quite certain it's the forsythia, but I would very much like to have a small piece to be able to identify it, because I've never seen it that big. This is
This is huge, yeah.
either it's several individuals that are fused, or this is one single individual.
Sorry, can we turn the lasers off? Yeah, just for
Sure.
imaging.
But then could you zoom out a little bit more so that we have the whole sponge?
Oh.
Look at that.
That's really nice.
Yes, this is maybe even 2 meters long, I would say.
It's very impressive.
Yeah. So bravo to the sponge growing in this environment where
Julio is
Coming down?
said that's a big one. Could we take it, please?
yes, yes, yes.
Well,
Do you want first to zoom in and look for your creatures or for other
Ah.
creatures?
oh, there were two. Yeah,
Maybe
there's a-
this.
No, not. Well, no, I'm admiring the sponge. I'm trying to see if any other critter is actually on it.
Ah,
But-
on the-
well,
on the
I
sponge.
see the whelk in front of it and this other.
Do you think this is a snail on the sponge?
The white one, eh?
Yeah.
You want the vortex lights off, the two big lights to see if it
Yes,
gives
please.
you a better picture there, or
Yeah.
try it and see. Best of
Yeah. Yeah.
Only better, or
So the star.
Oh, I like it. I like it a lot. So
Oh, look at the amphipod show there.
I know. I bring some colour to the game.
Yes. Look at it. It's so happy.
I'm not sure. The fish that was just in front of us just went by.
Could we have lighting from just one side?
yeah.
Could we try that?
Can we zoom a little bit for that? Do you think so?
Yeah. This is on.
Maybe just after the lighting
Okay.
change and then
Yep, yep,
yes. No, that's
actually for you to have it up to here so you have more. It doesn't
wow.
matter.
If it's not possible, no worries.
Yeah, that's one on there, but maybe a little closer, make sure.
Ah,
Are you
okay,
zoomed in at
okay.
the moment, or
Sorry?
are you zoomed in at the moment?
Are we
Nope.
zoomed in? Not so
No,
much.
we can zoom in if you want to.
Yeah, so it's-
Ah, okay. Oh, wow. Et voilà.
Okay.
Oh, that's
Can-
very nice.
Can I tilt it?
You know what it reminds me of? The intro of Mr. Bean.
Sponges. Yeah.
Yeah.
Oh,
No, it's not.
it's
No.
you had a little bit more.
A little lower. But... Sorry, can I play once?
Yeah, please, please.
I want a dramatic effect here.
We need to choose some dramatic.
Or it's going to be,
soundtrack.
you know, it's going to be taken by a UFO.
Like aliens.
Yeah, it's going to be abducted.
We just have a lock shift. Can I zoom in now?
Yes.
Please?
Are we back to auto for?
Yep.
Yes, we are. Okay. Yes.
We are.
This is very nice.
You're arriving right at the nice moment.
She did it on purpose. She wanted to. No, I think this is also very good for an
Yeah. So all the little holes you see are pores. This is where the water comes in or out of the sponge because it needs to filter the water. So the water needs to come in the animal so that it can feed from tiny creatures that are in the water, like bacteria or even pieces of organic matter.
That's why the Latin name is Porifera.
That's why the
That's why
Latin
the-
name of the
of the
whole group
sponges.
is porifera, meaning having pores. Yes, absolutely. It's still there,
at
the
this
fish.
Look at this fish tail to the left, to
Yeah.
the lower left. What?
Oh,
Well,
it's
well,
working.
yeah, no, it's hiding. It was.
It used to be there, yeah.
Yes. Okay. Have we exhausted our view
Yes,
now?
but do you want to look for?
We can for a second, but I think it looks good. But this is for a sponge person to sample now.
Yeah. So,
I'll go up to I'll send.
are you close enough to?
I'll need to move up a bit.
You'll need to
closer if
move
you
up.
want the
Yeah,
sample.
of course. Just a piece is okay with the arm.
Okay. Any particular side?
Let's see.
Where, as far as the sponge goes, just on the end?
Yes, on the.
Yes, not the tray, the actual sponge, but on the
Ah,
side?
yes,
Yeah.
on the side is perfect. Absolutely no particular.
Can I turn
Place.
the lights on again?
Yes. Did you already meet with the?
No, they weren't there,
Okay.
but they will.
Yeah. Did you see? I have my little blanket now. I'm getting cozy.
Yeah. So.
Yes, I should actually start to write.
Was 1160. Before 70, 1160. AC was at 60. Was it 70?
The Arctic.
Still swimming.
You said what genus did you suggest?
Forsythia.
Forsythia.
Changed.
I'm just focusing on that amphipod that is about to sit down there now. Just sit down. Sit down, please. I want to see you.
So it's 8 degrees. 35.
It's not.
And we are 1,244 meters below the surface. So I'm gonna write giant sponge.
Okay for you? The angle?
This is a beast. I mean,
Okay. Yeah,
this is probably the largest sponge
Yeah.
we've seen
Yeah.
so far.
Yeah,
right?
I haven't seen any larger sponge. How are we going to collect it? Do we do the manip slurp combination again?
I think they're going to try with the arm,
Yeah.
just to snip a piece on the side.
I would just be afraid that it flies off when we want to put it in the biobox.
So normally this species should be a bit more dense and should be able to settle in the biobox,
Okay.
I think.
Okay. I mean, you've sampled specimens before.
Yeah,
Of,
and
Can you zoom in? please?
yeah. I'm on it.
Okay, if you come back, I'll
oh, that's also a nice view.
Look at the little snail to the left.
Oh yeah, isn't that the one you sampled earlier?
I hope so,
Mm.
or I think so.
just look back.
It's a little bit orangey, you know.
It's very beautiful, and I love how it sits on the rock.
So I think we've sampled it before, but on the western side, that's going to be interesting to compare.
Not too close now. Should be right.
So what you have to understand is there is a pilot who pilots the ROV, and then there is another person that is actually working with the arm. It's two people. It's a two people's job. I will ask what they prefer, but maybe here is this here. What do you call this again?
Right or starboard?
Starboard?
Port side.
Starboard.
Ah, let it clear.
very misty.
Yes, yes.
Crinoids in the mist.
I really like the scenery with the carnivorous sponges in the back.
Yeah.
It's really nice.
No, I think because we're going to soon leave and the shift is going to replace us, we need to sleep. But maybe they'll have some interesting things.
Yeah,
up there.
it looks intriguing
promising.
back there.
Yeah.
Just a piece, Pablo.
Just a piece is perfectly okay.
Any piece in particular you
No, a piece from the side.
decide to?
Yeah, would be perfect. So sponges are quite resistant. They will heal easily from if we take a small piece, and they will regrow as well. They can regenerate tissue quite easily.
It's so spongy. I can already see
It is. It is.
I'm looking forward to that sample already. It's going to be nice to look at.
And I think this species also has interesting chemical properties because the specimens we've been preserving so far, they color the alcohol in which we put them,
Yeah.
meaning that the alcohol actually takes in some.
Big enough section.
That's
there, or-
perfect. That's perfect. Thank you.
Which stroke?
So we're thinking of starboard.
Starboard forward left.
Yes.
That's your point.
Yeah, I'm just watching you.
Tell me if you prefer that I.
Starboard forward left.
Yes, okay. Just curious.
Oh, that's perfect. So this is cast 6.
This looks perfect.
Most those off.
Yep.
Right.
Down.
Really nice.
Perfect. Well done.
No. Stay in there.
We're always afraid that
Yes.
they jump away,
Yeah.
but
We have had a tumbling sponge before.
yes, we've lost a couple of sponges to currents. No, this is perfect. Oh, that's a perfect way to end our shifts.
Yes, it's amazing.
Okay.
And to be honest, you almost do not see it that we took a piece.
Yeah.
I mean,
It's true. It's true.
really
Thank you.
lovely.
Thank
You're
you.
welcome.
That's over.
So,
That's over our arm.
oh,
It's locked.
Oh,
look at this. This is the device. How do you call this
So
part?
this is the master controller.
The master controller of the arm.
Yeah.
So you have a mini arm sitting on it, and this is what you're using to do all these really intricate movements.
And then our
For the
jaws, we can open and close our jaws here, or we can use the switch down here as well. So,
Wow.
it's really cool.
Thank you.
voilà.
Thank you for showing. I think we can continue the transect.
We can,
And
definitely.
So we're, I think, on the last part, the
Mhm,
last part of the transect. So we're good to go?
Good to go?
Yeah.
Lasers on, please. Thank you.
Got all the lights back on again, or?
Oh, yeah.
All the lights, yes,
Yeah.
back on again.
Yeah.
I really like that.
Yes.
I hope they will keep that image
Yeah, of course.
for sponges in the spotlight.
That's a very nice title for a paper.
Yeah. Yeah. I'll try to remember it tomorrow. But I've forgotten.
Sponges in the spotlight.
Yes, please, yeah.
Reach Alvi. Okay.
I'm imagining
we are
Julio
ready
on
to
his
move
bed
again.
watching the TV.
Direction
Because
is
he was
north.
probably not.
He texted
Yeah.
right away that it's a really big one.
So in our cabins, you have to know that we have a television, and we see what the ROV is seeing. So we can be in our beds and actually see live what the
Yeah.
ROV is seeing.
There are really many amphipods in the water column.
Yeah.
And did you spot the isopod earlier?
No.
There was a monopsid isopod in. Oi!
Look at that.
Camera out, please.
I'm just laughing because it's again.
Okay.
Yes, you will understand if you watch the episode on Loki's
on
castle.
Loki's castle.
Yeah, watch until the end,
Okay.
and you will understand why we're laughing.
Oh, this is beautiful.
Yeah, look at that. So someone was commenting earlier that this might be a new species.
The previous one we saw on this dive.
Yeah.
I'm not so sure about this one because it might be a different species.
Okay.
Can we go a bit down?
Yeah.
We can't see it anymore. Okay, okay.
Ooh, look at that.
Thank you. Wow. Oh. Stay with us, please. Just a bit.
Oh, it's going up.
Yeah.
It's going
I can keep coming up
straight
if you
up.
want me to follow it.
Yeah, maybe a bit. Oi.
Can you pan my camera?
Oh.
James?
Oh.
Oh.
Oh. Hello.
Wow.
Oh. That was very
Just
nice.
gone over the back of us.
That
Yeah.
was nice. Thank you for trying to,
Yeah, for
to
trying,
follow it. It's not easy.
No,
Okay. Well,
it's really challenging.
Nice.
yeah.
Paco, we have a.
Yeah. There's something.
Oi, this is beautiful.
That's a ctenarian. Ctenophore.
Ctenophore, and actually, we were kindly asked to sample them,
Oh.
Oh wow.
and I really would love to, but I can't really imagine how we would do that.
No, if we slurp them, they're
Um,
going to be. I feel they're going to be destroyed. I don't know if we have the right equipment for that. Wow, that was beautiful as well.
It was really,
Yeah.
really beautiful.
Was there a cartridge change a while ago?
No, still
No.
25 minutes because so our alpha died at some point. I think this
see.
might have
Yeah.
disrupted
Yeah.
the thing.
Gotcha.
Mm.
So the next team haven't shown up. We went down and checked, and they said that they're not interested. They're going on strike. It's too cold. So it's down to you guys for the next.
Well,
how many
as
hours?
long as we get more surprises like this.
They'll be here soon.
Yeah.
Oh,
I bet they would.
there's Sergi.
They came after all, huh?
Should we update you a little bit?
Yeah, let's have a little briefing.
Did you see the giant sponge? We just
That one?
sampled.
Yeah.
Monster. So as slurp goes, we have a complicata that I sampled at the very beginning. There's already quite a lot of sediment here. I should write it as well. So I think it's pretty full. This one, I think you,
Amphipods, right?
yes, with 2 amphipods. This one has a piece of glass sponge, but there's maybe space for
Yeah.
other things. Here, there's small stuff, tiny gastropods, snails, bivalve, and an amphipod.
Yeah.
And then it's free.
So number 3 is a bit more delicate than number 2, I'd say.
We've already taken a mug sample, and we've taken a round sponge and fluffy structures that are mysterious to us. There is also a mug, but it's not been used so far.
But there's a mug in this one as well.
Yeah, but otherwise, we just the big giant sponge.
The
There's
what? The one that
there's a
you just
piece
left?
here, but just one piece, so there's lots of space for other things.
Yep. Thanks.
Yeah.
Good job, guys.
So we're leaving our seats. Thank you for following us.
Have a nice trip.
Okay.
Couple of more squids. Morning, everybody.
Good morning.
And good morning to everybody out there in YouTube. This is Anna-Helena and Sergey taking over because Jenny and Paco
Yes.
are here. Are going to have their rest. They worked for 12 hours and now it's our time to do a little bit of work.
Good morning again.
We are right in the middle of the Transect.
Sorry.
Are we right in the middle of the Transect?
Wow, it's still kind of steep.
Yeah, come over to the right more. Come more right.
So, did they change state? Yeah.
No.
How long are we? 20
20.
minutes.
Yeah. And which sponges are we seeing, Sergey?
Oh, those are these exactinelids that we saw at the very beginning of the cruise. So something around in between 1,500 and 1,000 meters. Those are the ones that dominate. I think we've been collecting a few pieces of these sponges because we thought it was this Chaudinia
This is these are the ones that we have earlier been calling Roselida,
Rosellidae,
right?
yeah.
Yes.
So safest would be to call them Rosellidae.
And of course we didn't listen when we came down here to Paco probably telling everybody else what they're really called, right? But we will let him go to bed.
Yeah, there's some, you see this round stuff in here?
Yeah, look at that as well.
Those are Heodias. The one on top, the one on the lasers right now is a big Heodia. The one on the
But
right.
which one?
The one where the lasers are, this is a Heodia species.
Just give it down.
Which one? Sorry. I don't know.
Reach.
Probably
Can we reduce to
Heodia parva.
0.1?
So the one with the osculum in the very center, I think it's easier to identify, which is Heodia hincheli, but I couldn't see
And
his.
is that Asconema?
No.
No.
That's something crusting.
Yeah.
This blue uncrusting sponge, probably. Yeah, there is right on the right now a small ball, brownish. This is a craniella. The white thing, Reniera, I think we were saying. I mean, we need to put a proper name for this white roundish sponge. This is a vertical, one of these
Yeah.
vertical walls, right?
Yeah, which is not what I thought when I looked at the when I
Transect.
put out the transect on the map.
Yeah.
But that's the thing about going down there. You see nature and not a map of the nature.
But if you remember, we've seen already these walls covered, well, this is not entirely covered, of course, by these Heodia. They seem to like these vertical walls.
Yeah.
So again, this Heodia, I'm going to show you in a minute if there's any more appearing.
Reach ROV. Can we stop for now?
I think we get into the very summit. Oh yeah, these soft corals. guess same, yeah. It's quite muddy, isn't it?
Yep, it is. Quite sedimentary.
Yeah, there's of course this Lisendorix complicata. And this, you remember these Ascidians?
Yeah,
these
the-
Thiona intestinalis longissimus Ascidians. And you can see now, yeah, again, where the lasers are right now, on the right-hand side, you have these balls, which are like brownish balls. They're like leaving the image right now. Those are the Geodia species. Look at that. These small things here.
But that's like,
Mini spaghetti.
and that's bryozoa.
Yeah.
Lots of beautiful
Yeah, you're
bryozoa
right.
there.
But this is like a mat or something, right?
Yeah, I was wondering what that is.
Things that we're still in this vertical wall.
Yeah.
More Geodias, this Rosellidae with sometimes multiple oscula. This is Calophacus. This is where your amphipods are, right?
Yeah, you need to zoom in and check.
Oh yeah. Can you stop?
No, that's okay.
Yeah. Oh,
That's too late.
Where
We will
the-
get a new one.
Probably it wasn't a Calophacus and looked like Calophacus. No, this is one of these multiple oscula Rosellidae.
Yeah.
Just stop now.
Yeah. We thought this one was another species.
Which one, sorry?
The big one.
This one.
Yeah, but it wasn't.
Yeah, it's not. It is not. That's all right. Thanks.
Punch me on the
Group
screen.
check,
This
you
one.
can retrieve
Yeah,
the fish
That
eye.
one.
Okay, copy that.
Can you zoom in on the soft coral when we are here? Because they are kind of beautiful, aren't they?
Yeah. There's some legs appearing at the very back. What's
Which
that?
looks like a brittle star.
Maybe.
Yeah.
Ah, there's a serpuli.
Yeah. There are some worms there, or it's a
Ah.
little bit harder.
You remember this super beautiful dive from yesterday, the one in which we were like enjoying a small reef of serpulids. So you see at the very center of the image these tubes, these whitish tubes. Those are calcium carbonate tubes that are built by worms. These worms, they live always, of course, inside this tube, and they have a crown of tentacles that they use to get the food from the water.
Yeah.
Yesterday we saw one of these walls absolutely covered by this, probably this species. It could be the same one.
Maybe.
And
But remember, Sergio, we are in a slightly different water mass.
yeah,
It's 0.1.2 instead of 0.1. No,
that's
minus. Sorry, I am completely wrong. Minus 0.2 instead of minus 0.6. So it's a little bit warmer and a little bit saltier. So
So we are now in the south, in the northernmost sampling station so far,
but also the easternmost station.
and the easternmost. Yeah.
And this, I think, is a little bit more important, that we are on the eastern side of the ridge instead of on the western side of the ridge.
It's gonna take a beautiful shot.
Then we could please have the
Yeah.
lasers of.
Could you please turn off the lasers?
Yep.
Thank you. A tiny little baby of Germesia at the very left bottom side. You see now?
Yeah, this one?
Yeah, that one.
What is that stick there?
Oh. How about, I don't know, saying it's, yeah, I don't think it's a carnivorous sponge. Will be.
Okay. Let's move on.
Yeah. We'll continue with the
Continue?
Yeah, thanks, Will. So, is a huge specimen on the left hand side. This gorgeous sponge, Rosalidi. Look at that.
But what are those sticks there? Are they your very, very cool carnivorous things?
Not entirely sure. Yeah, I think there's a few of them. Yeah, so these branches that you see just hanging in there.
Yeah.
I think it was Avisocladia furcata. And this is really interesting, right? This is not muddy. On top of the rock there's a kind of rubble, right? What do you think is this rubble made of?
Oh, I don't know.
Look, this
That's a very beautiful gastropod.
super beautiful sponges. Lots of bryozoans.
Yeah, there are tons of them. I think they hang out together with the sponges here.
Yeah. Rhinoids again.
And your carnivorous sponges. And there is also in the background quite a lot of anemone.
Oh, Lysidendorix.
Oh, I haven't clicked that one yet. It was good to have my first Lysidendorix. Then I know that you are happy today as well. Yeah, good.
So our colleagues already collected a few specimens. Among those, there is a few amphipods. So I guess you also will be quite happy.
Oh yeah.
When the ROV is on deck. But we can potentially pick more. Of course.
Let's see if we see something.
Bridge ROV.
Okay, some craniellas in here. You see these balls.
Those little brains.
Yeah.
Okay,
Like
next move.
brown balls.
Direction 090 at 0.1 knot.
090
Oh, this is a carnivorous sponge. Gorgeous. You see that branching organism there, down there, and just beside a sea star, small sea star.
10 minutes until tape change. What are all those little white dots?
Yeah. I was just wondering this. I think it's a small encrusting sponge. They are really small and a little far away from us in order to be absolutely certain.
Where do you want to
Let's
stop?
You're still coming? I know we're on this side. Oh, look.
Is it
This
each
one.
here? I just need to.
Yeah.
I need a visual.
Yeah, that's
Yes.
the area.
You see these spaghetti, orange spaghetti?
The orange spaghetti. Are you hungry already?
Oh yeah.
You got up and you didn't go and have food before we started our shift.
Yeah.
Oh.
We'll have some time later.
Yeah.
But I think these orange spaghetti, those are the ones that were behind
Yeah,
the
they look the same, yeah. But let's check what they
soft
are.
corals.
So maybe they were not brittle stars then. But right now, nothing.
Oh yeah, I cannot see
for
now.
a sense.
I'm gonna go off. So how about these blooms? Are they easy to be clear today?
Sorry.
The-
These,
the-
yeah,
the
if I
blue.
put forwards on, but we're on a cliff face here, so
Oh,
I
yeah.
might thrusters on.
But are they going fast away?
Yeah. On the previous trials.
They will, yes.
Oh, yeah.
1 minute or less than that for
Yeah, thanks.
So we just have a little bit of a sandstorm because we had to, we wanted to break, to stop a little bit here, and then we have to use the thrusters a little bit, but we are told that this is going relatively quickly away. But if it doesn't, then we can continue, I think.
Oh yeah. Doesn't seem to be that fast. So 1200 meters.
1246 meters. Ooh, there something comes out of the fog.
Oh yeah.
Now let's see if we can find your orange spaghetti.
Mhm. Yeah. Curiosity, right? Oh.
There they are. Almost
yeah,
in the
yeah, yeah.
middle of the picture.
So, how about in the same sampling, we get this spaghetti, and we also get this beautiful. I mean, we're gonna get these.
You want to slurp
Reef
up
or
a little
whatever.
bit of the spaghetti and a little bit of the bryozoan?
Yeah, that would be awesome, because if you have a closer look in here, this is potentially bryozoan, dead bryozoan, right?
Yeah, it
Isn't
looks like it.
And there's an amphibot in here.
Yay!
Right?
Look at that beautiful eye. It's a coalesced eye. That is
So,
beautiful.
I hope
and
you
a
can
couple
see it.
of at the top there. Oh yeah.
Right in the middle right now, just below this snail on the left hand side, you will see a small amphibot. And I think we're gonna.
Do you see those two orange spaghetti? Will? Is it possible to?
So we would like to slurp them.
But we need to see them on the big pilot camera.
Yeah. So this community is interesting, right?
Yeah, that's.
This, oh yeah.
This, is it what it is? Yeah.
I can do the notes.
Yeah.
slurp chamber for
Yes, please. Slurp chamber.
Okay, I will zoom out so to get you on screen. Oh yeah. Oh, can you pull it a little bit?
this too.
Yeah, that one. This one.
Oh yeah. Indeed, this one. Oh, you're like. Can you pull it off?
George with the camera.
No. I was looking for that one. They were on the top.
That's perfect.
Ready for slurp?
Yeah. And there's some stuff also in here. This amphibot and.
Yeah, just.
Ready?
Yeah.
Oh yeah, beautiful.
That's very nice. If you now could go just
Can
a
you go
little
to
to-
the right?
Okay. Let's see here.
Yeah. So there's a few amphipods. There's a snail too.
Show already. There one.
To the left, right now.
Snail?
The snail.
Yes,
and there's
please.
an amphipod there. And
See that over there.
yeah, the amphipod is already
Let's
been
go.
served.
Okay, off.
Yeah.
Some stuff.
What do you think, Annelien? You happy
I
with it?
think that looks like a very nice sample of this. It's almost like a tiny little bryozoa reef, right?
Yeah.
Patches of
We done?
bryozoa for a long time.
So,
Thank you. That's great.
There's going to be plenty of stuff. So one of the missions of this course is, of course, taking samples, but it's characterizing different habitats that we find in here. This may be a microhabitat, right? One of these.
Yes, this is an
microhabitat that we-
interesting microhabitat, I would say.
And I don't think we've already seen this before. Probably we did at the very beginning of the cruise because we were not aware of these details. But now that we have gained a little bit of experience on the area and we have had closer looks at the different habitats, I think, we're like getting close to know how these habitats are characterized. Okay.
Time's your tape change up.
5 minutes to time change. Thank you. It was also very good to see what these quite biological mats are made of. It seems that they are actually tufts of dead bryozoa with living bryozoa on the top.
Continue?
Yes.
Thanks.
And it was fizz in a moment because we're very doing it. So we'll ask them to follow us up.
Yeah,
See what our way.
it's all right. So these small reefs or microhabitat are really important in the area because they create substrate for other organisms to live in, and I'm pretty sure we will have lots of tiny specimens in there.
Yeah, I think that will be an interesting sample for the gang who are looking at the small stuff.
Yeah, I'm pretty sure there will be plenty of these. So this is again the same.
Yeah,
Now that
it's
we know that,
beautiful.
this is a small reef of bryozoans in here.
But I'm also quite amazed at the biodiversity in these mats or reefs or whatever we want to call them.
Mhm.
These tufts. Can you zoom in on that thing?
Sure. I think this is one of these.
I think it's
I know
a
it's-
spider.
Ah, yeah. Oh, yeah. Can you stop a second, please? Oops. Oh, yeah. It's a quite challenging flight today.
Yeah. Well, it's right down there. So if
Yeah.
you zoom down in that corner,
Changed.
he's just hanging out there, right? Yeah, but it's going to be muddy again. We just wanted to check him
little
out.
bit. Yeah, so it's one of these huge Iguanodontids, these sea
Hold on.
spiders.
Is it Colossundae or is it something
That's a
else?
good question. I have no answer for this.
It's a beauty.
Two previous dives we collected
Two, I think.
two, and one of those. Oh, look, look
Oh,
at this
beautiful
amphipod.
amphipod. You are starting to find the
Yeah,
amphipods. Yeah, I like it.
Would you like
Well
to slurp in
done.
it?
No, let's
Ah,
not
yeah.
slurp it. Let's let it live. I'm learning the sponges, you're learning the amphipods. This is very good.
So we were saying that one of the previous pycnohonids that we picked was one that had children on top of it, right?
Yes.
The
But that was
one.
not the species.
Not this one, right? Yeah.
I like the amphipods sitting on the top there. Can we zoom in a little bit on the amphipod?
Yeah, I was expecting that we would keep going with that movement.
How much time do you have to?
You sure it's an amphipod? I think it's
You think
one
it's a
of those
shrimp?
shrimps. Yeah.
I think it's a shrimp.
Yeah, yeah.
Tape change just now, or?
Yes, it's in 1 minute. But we can move on and then we can do. There is another of
This
your.
orange
Yes, but
spaghetti, whatever,
yeah.
or small carrots.
You're really into food. I think you're hungry.
You see, this is Bryce one small reef.
Yeah. Let's just move on and change tape, I
Yeah.
think.
Do you remain stationary while you change tapes? Or are you happy for me to continue?
Oh, I am.
Probably do it here.
Yes, we can just hang here and then we can zoom in on that one and then we change after. Look at that.
Oh, this is, I mean, at least in our shifts, first time that we see one of those, right?
But now we have to change the tape.
Saying it's waving at you.
Yeah. So Dart says that he's saying hello.
Okay. Yeah, we need to change the
To
tape.
all of us.
Can I?
Yeah.
Stopped.
And these are the Bryce ones that we're talking about all the time
Yeah.
now. They're making this habitat. Reef.
It's recording again.
Thank you. Can we also radio the dry lab and tell them that we have changed?
So the B1 is the one that I took now from here.
Yeah.
But I think I have the C1 is the previous one.
So
Okay.
I'm not sure. Just check if it has
Yes.
anything or not.
We will check them both. Could
Let the
you
lab know, please.
let the
Dry
lab know? Dry lab?
That we change tapes. Thanks.
Dry lab ROV. We have changed the tape right now.
Well, we are
Alrighty.
changing as well.
Okay.
How big is it? It's not very big.
Can you turn on the lasers a second? It's not that big, right? It's star.
Sorry?
Do you think it's
No, it's not very
Not that
big.
big.
It's small.
Lasers as well.
could potentially?
See, if we have to zoom in to see the laser, zoom out to see the lasers. So I think it's
Yeah.
yeah.
10 centimeters.
So it's like something around 6 or 7
Ow.
6 or 7 centimeters.
centimeters.
You're good at that one,
Yeah.
huh?
Yeah, we're gonna take this out.
But where do you want to put it?
Reach ROV. Can we stop the vessel for now?
But then we're done with all the slurps. Are you okay with that?
I mean, if we can.
Yeah.
slurp some
Let's
things.
Yeah.
slurp this in. Can we then turn off the lasers again, please?
Yeah.
Going to have fun now. You too.
Could you turn off the lasers, please?
That's up.
I think this fits better.
So at the same time, we could also try to get these Bryce ones. I
They-
think they're gonna be quite
Broken.
brittle.
Yeah, they are super brittle.
Is for the same tank or
Next
the
one.
next one?
I think the next one, yes.
Is the last one?
Yes, that's okay.
We have the bio box on the right, still empty, right?
Yeah, I think it's fairly empty. I wouldn't. piece of sponge.
Yeah, sorry. So one of the objectives of this, when doing this sampling, taking these casts, is taking samples that help us identify the different organisms that we're finding there. If we get these samples, we're going to be able to study them in the lab, doing morphological studies and also, very importantly, genetic studies. Yeah. Can you also zoom some of this white stuff in here?
White stuff, yeah.
Because those are the
white
brightest
stuff.
ones.
Just
The
small
thin,
branching.
branching ones, yeah.
I think you slurp
Lace.
them for, just take a step back so that
Yeah.
you don't slurp.
Okay. I should just come back a bit so you're not taking the hill.
Yeah, that one's beautiful though, the one on the back.
Shout when you're ready.
Oops.
It's got a lot of things.
Yeah.
Go for it out. Okay, on.
Yeah, it got in. Thank you, Antonio.
There's some stuff coming in here, right?
Yeah, I'm sure that's good.
Started. She was talking like that.
Oh yeah, start definitely. Yeah, this stuff is just.
Did you? Yeah. Notice those. So yeah, interesting.
Thank you.
Good job, guys.
You guys tried to slurp the sponge?
Yeah,
Yeah. Starfish need it, no?
that one, the big one. Yes.
No, not the big one, the small one.
Ah, the small one. Okay, yeah.
So that's one.
Yeah.
So coming in from the dry lab is the voice of Julio, our
This one here, yeah.
third sponge person on board. And he is extremely interested in all this small stuff that nobody have looked at before. So I think he already has a little
Yeah.
group of possible new species on board
Yeah.
here.
Yeah.
So Julio, that one
Thank you.
was for you.
Okay. Maybe he's just come up a bit clear and I'll kick it on again. I'm sure it's some quick through.
I haven't seen it. Or is something just dropped in there?
Getting our quick kick?
Yeah, yeah.
Okay.
That's a beautiful crinoid sitting on top of the fan-shaped
Yeah, it looks like it's
sponge there.
You have it?
Yeah.
Oh, wait. Can you reach that thing over there? Yeah.
And when you move the shoulder
This one?
down,
And those.
Is it too far?
Something more?
To go
There-
down.
Start as well, yeah.
Yeah.
Because you saw, I feel it
So-
coming.
Above.
Sergi has spotted the Lysodendorix complicata in the picture. I think we should almost have a game, spot the Lysodendorix.
That would be a really easy one, right?
Yeah, that would be a really, but it's in the middle of the night for us. We do like really easy games in the middle of the night.
Yeah.
Might be better off coming to the
I
right.
think they also collected a snail, probably this one, right?
They already.
Uh,
I'm just having the space.
Alright, use your...
Otherwise, I'm pushing off.
Shout when you're ready.
Oh, the crinoid was not very happy with the sampler coming, but
John, Go a little bit closer.
For the cake.
Yeah. It's not wanting to come off, is it? That's
There.
That's a
Yeah, no, it's just okay.
Yeah, thank you so much, guys.
Well, that was
And the anemone also
Really clean.
curled itself in a little bit, so
yeah.
but it will come out again.
So the idea is that we need to sample some of these organisms in order to identify them properly. And the idea, when especially when in a habitat like this, which is like quite challenging for pilots to be sampled, is to get as many specimens that we need in each of the landings that we do it. This way we maximize sampling and take advantage of this landing.
good. Okay, happy.
Yep. Thank you so much. Can you turn on lasers, please? Oh,
Yeah.
This is on.
This is on.
It's too long. So I'm feeling comfortable at that height.
Okay.
Right, okay.
So we are going to try to continue along our transect a little bit. But this was a nice little stop to check out that community that we normally just see as brown blur when we are flying past
Yeah.
them. And there are so many nice details when you go into even these things that look from a distance possibly slightly less interesting. And then we zoom in and we see that they are super interesting after all.
So we're mostly focused on sometimes on this megafauna that we call megafauna, which is like this super big or big
Bigger than
organisms.
10 centimeters?
Yeah. You can spot them with in the distance, but there's a lot of
Beautiful
more-
biodiversity
yeah.
in the small parts, right?
especially in this reef, but also in the sandy bottom. So you've been sorting a lot. These
Oh
days.
yeah. I spent my last shift just sorting the sand. That sounds fun, right?
Oh, this
It
is-
was actually quite fun.
I think this is the reef of the small reef of the serpulids. So we have two. This brown, lighter brown. This thing that you see at the bottom. This is the bryozoan.
Mm-hmm.
Yeah. And then the one hanging on the rocks. This one in the middle of the lasers. I think this is also built by organisms. So this is a calcium carbonate remains of these serpulids, these worms that we just mentioned. Oh, look at that. So we are reaching the very top, I guess, with this soft corals. A huge sponging here to the right, where you can see a lot of crinoids. Beautiful. Seems to go a little bit down here.
Yeah, it seems to be a little bit of a down and up again. And since this is seen more from a distance, we again see mostly the big things
Yeah.
from afar.
Which are these Roselidae. This big thing in there is white, I think. Is Endorix, and then I think there was one of these beautiful and big fan-shaped sponges.
Okay, can we start moving direction 120 at 0.2?
Can you stop right there in front of it? We're gonna take. Is that the Lysodontorix?
But now you have to put it in the bio box.
Yeah, it's attached to a rock.
This one here.
The one on the back. Here. And then we'll have a look at the small things around this habitat, this sandy habitat.
So the reason why Sergi wants to sample the Lysodontorix is because he wants to compare the genetics of these Lysodontorix sponges, the spaghetti sponges, to the others we have sampled previously during our cruise. Because Lysodontorix is brooding sponge, so it doesn't disperse very widely, but it is very widely distributed. So that's a little bit of a conundrum to find out how can they be the same species? Are there very separate populations in that and stuff like that? So that's why we're on every single dive talking about Lysodontorix. We know it's a super widespread species.
Yeah.
And very common, as you have seen, because the game spot the Lysodontorix is not a very difficult game.
Scoop. Probably. Scoop. Empty, right? Yeah. Well, not empty,
But I think you can go for starboard, front, right.
How about the what's the mark here, or we can
Bridge
just wait
RV
until
will
there's
stop
another
for
mark
sampling.
here.
Can we stop the vessel for now?
We
Sorry.
definitely need to
Yeah, we
take
do.
two, right?
Yeah.
So this is going to give us the opportunity of collecting the one sponge that you see on the back. Well,
Take this sponge out of the tray. That's the objective.
you will see where the lasers are right now on the left, and we're deciding whether to grab the sponge with the rock, with the arm, or just using the scoop.
Is it on a rock like, or
I think it's on the rock.
Do you want to zoom in and check
Yeah.
that?
are you able to just grab the rock if it's small enough? It looks like it might.
I mean, look at that. It's 10 centimeters wide. What do you think?
Might be able to.
Or just scoop it if you. If
Yeah,
you
either or, whatever you prefer, yeah.
And there's some let's just scoop it
Scoop, yeah.
because there's some serpulids in there too, so we can just study.
Some
So you
wormy
see?
stuff. Yeah,
Yeah.
you want the worms.
And there's some small sponges too. So this is the one that we're trying to collect here now, using a tool. Really efficient one that we just used in the past so many times. Yeah, you will see it. This is the scope. Well, this is the arm trying to get to the scope, and you will see how it performs, which is like a shovel, right?
Yes.
Yeah, I'm gonna.
Zoom in a little bit just to see.
Yeah.
if it's-
Can you see it? Because I just see this blue all over the place. Yeah, I think it's. Yeah, that's It's in the right position, I think.
Got it.
Got it. So it's gonna be top right, this one.
Top right.
Because there's something else in the other one.
Okay. Are you happy with that? You okay?
So the stone on the
So you can start here if you want.
start just below it.
Yeah, because there's some.
He wants the
small
white
sponges.
little
Yeah.
squigglies. So here's a little bit of a, not really a secret, but a fun fact about Sergi.
Uh-huh.
Before he became a sponge man, he was a worm man. And I think
You want it?
worms are still very dear to you, right?
Yeah.
Already here is
Yeah.
is it better? Yeah.
So the combination of worms and Lysodendroics are just, well, you're just
Best one.
sitting here and smiling like crazy, and you haven't even had breakfast.
Oh.
Look at that. Everything is coming. It's beautiful. And you also got, is that the brown asconema?
It could be one of these small ones.
Yeah,
I think
Or-
it's attached to the stone, so
that's very good.
which draw for this one?
Top right.
Right.
Top,
Please.
top right on, yeah.
Yes.
Yeah, can
Yeah,
we zoom?
yeah, that one.
Can we move so he can see?
Can you give it? So it's backwards.
You should try to open a little bit more.
Yeah.
Oh. So we need to know
Yes,
that there's
All right.
Parts just fell into the.
Yes, that's
fine.
okay.
We took note of it. So there's a forsythia in there, which is easy to. It's gonna be easy for us to identify which one is one. There's a really beautiful specimen. Plenty of other
is.
things you will see. There's plenty of crusts, and on the other side we find
Your
some of these
worms.
units. Yeah.
Your worms.
Oops. Yeah.
Woohoo,
Absolutely.
in it goes, and in comes
Might still be
also.
some of the worm stuff.
Oh,
Just keep
what's
shaking
this
it
then?
up.
Yeah.
Yeah, please shake it. Shake it, baby, now. Twist and shout.
Good. I think it's good there. Thank you
Thank
guys.
you.
That was awesome.
Is that all?
Yeah,
Yes,
for
sir.
now. Yeah.
Okay.
Thank you so much. Just check around. Oh well, I'm gonna need to follow to zoom in order for door to put this scoop back. Okay.
In it goes.
It's good.
Great job, guys. Let's just look around.
Yeah, let's just zoom in and see what it is
Okay.
there.
You were saying, what is this small white stuff? Probably small sponges, right?
Yeah. Now I am not surprised anymore. I'm not surprised of the
And
morning.
you see these tubes in here? Those are forams, right? Or
Oh, maybe, yeah. I think so. I think there are a couple of foraminifera here.
elongated tubes, brownish. As you see at the very bottom, those are forams, which are unicellular organisms.
Which
So
is
they
kind of
can-
crazy.
But they can be like huge structures.
Yeah,
I mean,
but they
compared
are conglomerating. So
Yeah.
they're just gluing themselves to each other.
Yeah.
Bzz, and then they're stuck.
Forever.
You happy for us to continue?
Yeah.
Yes,
Yeah.
we're happy to continue. Thank you.
That was a really productive sampling, really productive stop.
Yeah. And maybe zoom out a little bit. And let's fly and see if we can find some
So we are
other stuff.
on the right hand side of the Transect right now. Some small things.
Yeah,
there,
but-
here and there.
Again, when we looked just now and saw it's more diverse than it looks from
Mhm.
above, right?
Big sponges on the left hand side. Probably this Roselidae.
That is the one on the right, right? Yes. I'm happy with
The potential to try to get back to the original one. Yes. That direction. What do you think?
Yeah, we
We are
kind of, yeah,
at the right hand side of the
yeah,
Transect.
maybe to get a little bit back to the edge, but not over the edge,
Yeah,
kind
okay.
of. So our transect is planned as flying along the edge of this unnamed sea hill, sea mount. And see what we see on the edge there.
It's a fish.
And we are now a little bit into the more flatter part of the sea hill.
1,228
Reach Alvi.
meters depth.
Yeah.
Okay, we are ready to move again.
This is the ring
Direction
of the
120
Serpulid.
at 0.2 knots.
We 120 at 0.2.
Yeah.
But it's quite a lot of food in the water, I would
Yeah.
say. So it seems to be a place where a lot of energy is coming from above.
Yeah. That's another fish.
Yeah, that's another fish. Well spotted.
Roselidae.
With a crinoid on it because they do like to decorate themselves.
Mm.
Sorry.
Yeah. Okay. So. So dive was started at around something around 1,500 meters. there was a really steep part of the Transect at the very beginning.
Yeah. Then
We
it
were-
was up
We were
this.
sleeping right at that
Yes.
point.
I think you all out there, you saw more of this dive than we did because we just got up from our beds. It is at the moment almost 2 o'clock UTC, and we are at UTC +1, so it's 3 o'clock ship time in the morning. So our shift starts at 2 in the morning and lasts until 2 in the afternoon. So that's why we are talking about breakfast and stuff like that. Again, here are the beautiful
Ooh.
Roselida
More life in here.
decorated with crinoids. Or maybe we should say the crinoids are just using the Roselida as a stage to get a little bit further up into this very nutrient-rich water.
It's mostly
All of the
Roselli, and that's
small
it.
little balls. Oh, and spot the
yeah,
Lysidendorix.
We will pick another one, but not now.
But can we soon have a little bit of a game with, a gift for the people who spot the Lysidendorix first?
Oh yeah.
So we can see who in the shack will win the game? I
What was that?
think maybe Will be the winner of first spotting the
Can
spaghetti.
we change the direction of this movement to 080?
080.
There it is, an anemone on that rock. A white and pink anemone.
So these Lysidendorix, I just saw one. They're not that abundant in here compared to other places, but they're like quite big when you find
Yeah.
them. They're like gorgeous.
Well, they are very pretty. And the red thing there, is that another...
Sea star, yeah.
Is there
Let's
another
just
sea
check
star?
in the distance.
It's another
Ah.
sea star.
But probably the first, the same one that we picked already, right?
Yes.
Yeah. It's difficult to get the image when we're flying. But yeah. I'm quite sure what's the one. So we're trying to reposition the ROB in the Transect so that we can follow more or less the crest of this very summit with potentially more rocks and more life in there. And we forgot to do the mug.
Yeah, but we can do that later.
Yeah. So you can probably, well, whatever you feel more comfortable about, explain what is the mug.
Oh, yeah.
This mug thing.
We have a coffee mug. Not a proper coffee mug, but an equipment that looks like a coffee mug. And a metal thing, and it has, instead of a bottom of the mug, it has plankton net, so a very fine meshed net as a little bag hanging under the mug, so we can use this to sample soft sediments. And as quite a lot of us wanted to find out about all those little animals that live on and in those sediments, this is a nice little tool to bring back to us some of the sediments. And so I said I had been sorting sand and mud the entire last shift, and that was samples from these kinds of this kind of equipment. And the animals that live on, and especially a little bit down into the soft sediment, are little crustaceans and little worms and little mollusks, mainly. And when I say little, I mean 2, 3, 4 millimeters maybe at the maximum. So that is all work that is being done under a stereoscope, a sorting microscope, sorting stereoscope. It's nice to do when we have no waves or flat seas like we have now. It's not as fun to do when the waves start to get there, because imagine you're sitting at, say, 40 times magnification, and that means that also the waves in your Petri dish are going to be 40 times magnified, which is not always a good feeling. Seasickness-wise. But we are very, very lucky on this trip so far, and have had super beautiful weather.
Yeah. Hopefully we'll continue until the
Yeah.
end.
Well, let's see. But we just have to be happy with the weather we have had until now.
Yeah. It makes things super easy, or easier. It's not super easy, something in here. But yeah, I mean, this flat sea, it's something that you wish at the very beginning, and I thought it will never will happen eventually, but not for a long period. And it's been like this for a few days.
Yeah, it's been beautiful. And remember, we're relatively high up in the Arctic right now. We're at 76 degrees north, which is way into the land and seas of the midnight sun.
How much is it soaked?
So we never have, it's never dark anymore here.
Yeah, no, I just want to know.
Difficult sometimes to sleep.
To know how much
Yeah.
it's soaked.
You think so?
Yeah.
At least for me, yeah.
Yeah.
No.
But you are
No,
our
I'm just
southerner
asking
on
a
this trip.
question. I'm bringing it
Julio
around.
is also.
Julio, 30, and I would say also in our ROV team we have 2 pilots from Portugal, so that would also be southerners.
Yeah.
Yes. Welcome to the north.
Yeah.
Woo! We see right into sponge again.
Oops.
So we are on this soft sedimented area, and we needed to use a little bit of thrusters to move, and that's why we've had a little bit of a cloud. Should we maybe... Oh, there is a curled up beautiful little fish. Oh yeah. Sleeping. Well, it's the night, so I can understand that. I was also very nicely curled up in my bed not too long ago, and that was very nice. Is that another fish there, or is it? Yeah, it is, right?
In similar position as the one.
Yeah, it's the sleeping position.
Yeah, look at that.
That's even more curled up than I am in my bed.
that's another
Ooh,
one
it's
there.
the sleeping area of the fishes. See, curling up.
That's good.
And another one.
Another one there.
Wow, we're really into the sleeping fish area.
Do you want to jump in?
Yeah,
there.
another one
So
there.
a little bit forward on.
Do you think it's something in the water?
I don't know.
Nah.
So a little bit of ups and playing on the joystick to keep it.
Alright.
Going up a little bit. I'll tell you what I'm saying.
Ah, you got all man.
I think we got
And there
to
is
the other side.
there is a straight fish sleeping
Ah, but
outstretched.
another one there. You see?
Oh yeah,
On the
curled
right.
up, and back there was the outstretched sleeping
There's a lot of
fish.
insistence.
That's a stalked sponge, isn't it? Ah, no, sorry. That was one of these Raniera or whatever.
Reniera.
I think so.
Name here. Quick name here.
90, yeah.
Oh, but there is a non-sleeping fish.
That one was really active.
Yeah, well, he just
Yes,
woke
yes,
up. He
yes.
had a little bit of a sprint, and then it's the interval
There's
training,
another one
right?
there. So there's plenty of those.
Yeah, look at
Fishes.
that.
Go check the dry land. You put out the 6-man camera for a while. There's lots of sleeping fish in there.
Yeah.
Okay, coming out.
Anna, so you would you like to drink?
No, thank you.
Okay.
We're good.
Thanks so much.
Thank you. Okay, so we are at the Sleepy Fish Grounds.
Look at that. Yeah.
And that is our Sexton camera coming out.
Yeah. You see it. Yeah.
So we're trying to
Yeah, trying to come back the way. Yes.
So maybe we go up and out.
Straight one.
Yeah. And there is another straight one under the rock there.
Put my head a little bit more.
That's
Yeah,
another
that's a
species,
different
right? Yeah.
species,
Turn off the
yeah.
laser, please.
Hey, lasers off.
There are some swimming fish over there as well. And a beautiful snail.
I don't know how many fish so far, but a bunch of them, right?
Well, maybe we already passed the sleeping section.
Change the to north. Bridge ROV, can we change the direction of this movement to north?
Maybe to north.
And increasing the speed or
Yeah,
and maybe increase the speed to
It's a sleeping
0.3.
one.
Yeah, there's a sleeper.
North at 0 point.
Okay, that's it, thanks.
There's another one on the right hand side,
Yeah.
sleeping.
And what is this sponge?
This is the Rosellidi.
This is the Rosellida. And some fishes in the background.
mix.
Okay, I
With
think
rock.
you are winning the prize today. What will the prize be?
I don't know.
An apple?
Yeah, or a beer.
Should be able to do it today.
Okay, yeah.
You had on the last 3 days. You are getting used to having every day.
It's a good movement. So is that the fan-shaped thing?
I think so, yeah.
Yeah, fan-shaped sponge.
Fun shape, brownish.
And a sea star, pretty one.
We don't see many of those stalk crinoids that we saw in previous similar settings, right? Like a sedimentary.
Oh, but this is a little
Yes.
bit more coarse sediment,
Yeah.
I would say.
So it makes
It's
a difference.
more gravelly sand instead of muddy sand. And that makes a big difference.
Yeah. It might seem same or really similar to other habitats we've been, but I mean, given the fact that there's like this coarse sediment, yeah, you cannot find the same species. So we are not finding the same species.
Is that a rock or a hole? That's a hole.
Yeah.
So it's this semi-hard sediment,
Yeah.
which would again support a completely different fauna. Oh, what's on top of that rock there?
Crinoids.
Or is it?
Basket.
It's basket star,
Can
isn't
you
it?
stop here? Have you? Yeah, because we can have a look at the
Yeah, that would be nice to have a look at.
beautiful one. But somewhere here, so that we have it. That's gonna be a beautiful shot. So you will see in a minute this creature. This is a
Basket star.
bizarre, right? Creature.
I love them. They're
Yeah.
really pretty.
Echinoderm too,
Yeah.
ophiuroid too.
So in the group of the brittle stars.
Yeah.
But the arms are branched like crazy. Oh no, no, no. My nose were because I was pressing the wrong button in our logging
So Dorota
system.
and Antonio are trying to position the ROV just in front. Oh look, a fish coming to the picture. Well, it would more closer. Yeah, this will be a super beautiful image.
Yeah, that fish is searching for food along the seafloor there. That's great.
So you usually come there to check out
Yeah.
the chest.
So this rock is
Pretty.
really beautiful.
We are going for pretty rocks at the moment.
Bridge ROV, can we stop for now?
So something to. So we're gonna try not to burn the image with light. So we like doing some changes.
Change the lights to see if it.
Yeah,
Yeah.
it's too bright.
Oh yeah, that's better. But this is a rock that has most of our
Yeah.
nice megafauna. Ooh, look at that.
Oh, that one.
That's good.
Yeah, look at that. Zoom in a little bit. Ooh.
I don't know about dry lab if they like the image or whether we can do something else.
But we like it.
We can like it, yeah.
Yeah.
Slightly to the left.
So
Is that okay now, Daniel?
A little bit more so you get the phone shot to the left. That's good, thank you.
we have
That is a beautiful one.
the producers of this live stream are sitting in our dry lab, one floor, one deck up from us.
Let me take a picture myself.
So you are just positioning this to have a nice picture. It is a beautiful picture. Maybe I will also take a picture of this. And the pilots. Then you know it's a good place.
I was taking some holiday snaps.
So this fish just came to the picture.
Yeah, well,
to be
maybe he
present in
wanted
the
to be
picture.
part of the gang. So tell me now, we have a soft coral,
Yeah.
we have a basket star, we have Rosellide,
Yeah.
we have crinoids, and we have fish.
And then some small stuff also in there, which are the
Serpulida?
Serpulidi, yeah.
Yeah.
Uncrusting sponges everywhere. So
Oh
thank
yeah.
you.
Okay, so I will start taking notes when you are enjoying this beautiful thing.
So if no one says, I'm just going to do a close-up in the basket of your right, so that we can see more details on these organisms.
And now just let it be. That's beautiful. So he actually uses most of his arms to cling to the rock, but look, they're moving all of these little parts of the arms. I'm always amazed at. I mean, I can sort of control 10 fingers, and I tried to learn to play the piano when I was younger.
Nope.
And
Now
it was kind of difficult to coordinate those 10 fingers sometimes in the more difficult pieces of music. But imagine coordinating all of those branches of arms.
that is a beautiful one. And these are, these like small
they're
things that you cannot
great.
imagine that exist. These eyes exist, and there's different species in different areas. I come from the Mediterranean, and there's it's quite frequent that you get these organisms in around 150 meters.
So you are a little bit shocked to find it at 1,220.
Well, probably, yeah,
It is quite,
they're common, right, at these depths too.
yes.
So it depending on the species and the habitat, they can be some shallower depths or deeper depths. But this one is really beautiful. Yeah.
Imagine playing the piano with all of those fingers. All of those little tips of
I'm gonna
arms.
make like as close as it can be. Well, this is the closest we can we can get. You see the small
Tiny little
structures.
branches
Yeah,
of,
they're moving.
yeah, all of them are
Little bit.
moving and doing little things.
Okay.
Very often we find these also clinging to structures that are sticking up of the sediment.
Yeah.
If you go a little bit shallower to where we have the big sea trees, the bubblegum corals, we very often see them sitting on those as well, waving their arms around.
Yeah, they're moving the arms like really slowly.
They're playing a slow sonata.
You can see it now. Not the fastest organisms in town. But
Oh, but
I mean,
also not the slowest. Come
no.
on. You are working on sponges. I don't
Yeah.
think you should talk about speed here.
Well, there's some sponges that have been identified to be moving. I mean, they move like take years for them to move.
Yeah, right? Yeah, so in that race of the turtle and the hare, they would still be the
Less than the turtle.
less than the turtle.
So yeah, this basket star. Well, what do you call it? Basket?
Basket star.
Basket star, yeah.
Gorgonocephalus. That is, of course, from the Greek myth.
The one.
So some people also call them the Medusa sister because they are like the hair of Medusa. But yeah,
So
pretty. But we can move on.
Yep.
Yeah.
Gonna resume the flight.
The lights back up again, yeah.
Yeah.
Yes.
Just try to get back to the.
Back to the light.
Yeah.
It's cold in here today, right?
We meet some people.
looks like.
Some people around. We meet some other people like producing heat here. Oh, yeah.
We will resume our flight. After this short view of the Gorgonocephalus.
Same speed, 0.3. Bridge, are we?
Bridge.
Okay, we are ready to go. Next movement, direction 350 at 0.3 knots.
That was a sea star we didn't see.
350 at 0.3.
Okay, that's it.
So it's a 1 hour take.
There is another sea star.
Can you put on lasers, please?
Lasers, yeah.
Yeah, laser on.
So this is a quite pebbly ground, and it's really obvious no more soft sediment. I mean, there is a little bit of sedimentation on top of this, but it's not really soft anymore.
Yeah.
Is that another of those biological tufts.
Could be,
Yeah.
yeah. So it's getting more spongy.
Yeah, well, it's harder, so
You can tell. In the distance, you start to see this rosellity coming up. The dominant species, or maybe there's a few species, 2 or 3 species. Also. This is the reason why we are not putting a name on those, because sometimes, in order to be certain on which species we are seeing here, we have to pick samples. This is the reason why we stop sometimes to get these samples. This is
that's a
cute
beautiful.
one. You see this one? More fun shaped.
Yeah. But let's go on.
Yeah. Thanks. You see this one? It's a little bit different, right? With a kind of stalk there.
Yeah, it almost looks like it has thin start and then a wider top.
Yeah. Is it one of these fun shaped? I don't know. Yep. Yeah. Nice. Yeah, we're getting close to the original transect that we planned. I think it was Jenny
Yeah.
planning this.
Jenny
Transect.
is our, together with Julio,
Yeah.
our transect planners.
So no more fish sleeping in here.
Well, we can't have sleeping grounds all the place. There need to be, places where you sleep and places where you don't sleep.
I know it's okay.
But is that a fish?
Yeah.
There, yeah.
We're close to the original transect now,
Yeah.
and I think there's like a
A crest again,
Crest.
right?
Yeah, that seems to be the crest here.
oh yeah, we can just follow the
turn
crest.
and face.
We can just go this way.
Yeah, because if not, we're going out into the great unknown.
Yes.
This is how we're getting. Oh, yeah.
And also, of course, having a look around and see which.
Yeah, because this is a Rosellida
Strike is
place
amazing.
more than a Lysodontorix
Yeah.
place.
Definitely. Let's check.
Is that another? No, I'm pointing too far down.
Carnivorous.
Was it
Oh,
carnivorous?
yeah,
Wasn't
I
it?
think so. Yeah.
Well, We haven't talked about the carnivorous sponges for some time because I think they are still a little bit, they're going for my amphipods.
Mm-hmm.
They're not giving me nightmares yet, but they are a
There's
little
plenty
bit.
of amphipods.
Yeah, but they're all cute.
Mm-hmm.
And there aren't a lot of carnivorous sponges right here, so this is
Yeah.
a good spot. But that stick there in the middle of the picture
Yeah, I think so.
is a carny sponge.
Potentially, yeah. Can you get bored?
Where's
Look at
Harbour?
that. Yeah, because this is the right, the left hand side is the kind of the wall.
So this is the thing flying around, and it's large, but it's compared to this mountain relatively small.
Yeah, sopranos.
ROV and mapping, sort of finding out about the mountain, just with a general bathymetry, and then detailed checking it with just the ROV is like mapping a big mountain just with what you can see using a handheld torch in a way, right? On land. So it's really all those
You start-
finer little
Can you stop here?
details of the ups and downs and hills and crevices and everything on the mountain we just see for a very little distance at a time. I think Sergi wants to sample the Lysodendroics that
Yeah.
he's seeing in the picture.
So we have two.
One big
This
and one
one
little.
cannot be picked with the rug because the rug is bigger than the boat, right? How do you think we could potentially? You think with the you think?
Straight off, yeah.
Yeah.
But do you really want to do the push course now, or do you want to do it on the next dive? Then we will have better processing time.
please.
Next time, yeah.
Mark?
Okay, cheers, Paul.
Once we
Yeah.
find another place.
Because which is half an hour.
Then we should get a few more samples. Go to the right because there's more sandy habitat. Take the map and put it on. Right? What do you think, Christian?
Yeah. So in the background here we do have some carnivorous sponges. Up
Just
there.
have a look. So here and here, right?
Mm.
Oh, yeah. Give me a sec. I'm gonna take a close up on these. Yeah, potentially, yeah. This thing in the middle,
Yeah.
Ranchy. Check. Okay. So we could do live stream after their live stream, we can still collect some stuff. Yeah. So, do you think we can use the scope to scope these one?
Yeah. I'll just creep forward a wee bit as well. We'll have to bring the sextant in as
Oh,
well.
yeah.
Yeah.
We have to, yeah. Daniel, we take in the sextant now because we are sampling.
Yeah, that's fine.
Thank you, guys.
So this is not the easiest place to land because it's
Ah.
kind of steep here. But as we've said before, we have wonderful pilots who can do the almost impossible. They make the impossible possible.
It's not impossible for you guys.
Bah.
They start with draw now, yeah.
Yeah.
Antonio is gonna get the scope in order to get a nice sample of Lysandorix complicata. Zoom out a little bit.
When you start withdrawing again, okay.
And you want to have it in starboard and then
Yeah. Well, let's
one
see
of the
where it
backwards.
gets when it gets.
Yeah. You still see it.
Yeah.
That's
Nicely done.
just rotate a bit and try and scrape off the edge. Yeah. Just try and rotate and scrape.
Beautiful.
Just scribble away. Take left and right with your wrist on the shoe. Just like that. That's it.
That was super clean.
Oh yeah, that was nice and clean, huh?
Nicely stored.
So no carrot.
Sealed or stored and properly hands.
So we would like to have it in the starboard, one of the two.
Starboard, okay. Just give it a shove now, yeah.
They're placing both.
Okay.
The ones in the back.
Yeah, both of the ones in the back should have space,
Okay.
and whichever is
Beautiful.
best of you for you to. Okay, it's going to be 7.
Okay, that one, just on the edge. Rotate the cable.
Yeah. Beautiful.
Just make it elbow. Lift it.
Awesome. Thanks, guys. Again. Very good one. That one there.
Which one is it?
So we
Also with
will.
the scope.
Yeah.
scoop another little sample,
Okay with the drawer out.
and with that sample we are going to end the live stream of today. So enjoy your last Lysodendroics, and since we all have spotted it now, we can give ourselves an apple or something like that to celebrate that we have all seen today's Lysodendroics on a dive. And we will be back to you. So follow this space, I think is what we should say. There will be new live streams coming.
Yep.
But ooh, see, there is an amphipod swimming.
Mm-hmm.
Yeah.
Can be
That's a beautiful thing to end the live stream with. But we have to look for them.
Yeah. And then potentially we can also have this.
Let's see
Rise
what
one.
happens.
You see them?
Yeah.
Back?
So Sergey does not have time to say bye to you, but I will say bye to you all from both of us.
We've got quite a bit of pitch as well under that yaw, you can still use it.